All righty. Good morning. Today is Monday, August 31st. Let's go ahead and get started. Any questions before we get rolling today? So done silence, always what I enjoy first thing on a Monday morning. All right, let's talk game plan then. Today we are going up and moving up to our second level of the hierarchy. We have done everything we needed to do with chemistry. So now we get to start talking about cells. We will start first by talking about the anatomy of our cells and its general function. And then we will start talking about our physiology, including our very first major physiological process of the cells, and that is the process of mitosis. We still have a handful of assignments due in this section. On Wednesday, your Unit 3 review. And again, remember those review pages, questions are mixed in with all the activities at the top of the pages. Again, you obviously don't have a microscope, so you can't do most of those activities, but you do need to answer all 10 of the review questions on that. Also, your second labster, mitosis, is due, 80% correct, and you must complete the entire activity. Then we get a holiday on Monday, come back on Wednesday, and we have 3 more assignments due. Your Unit 4 review is going to be due, your PhysioX Exercise 1, your second type of lab simulator. And remember, you're going to get 5 different activities, so 5 different lab reports that you are going to be turning in, and then another labster. Also on Wednesday, because it is the day before the exam, we will be doing a practice lab exam, so you can see kind of what the questions are going to be like and what that format's going to be like. It's not for any points. You won't be using Proctorio for that. I'll answer your question in just a minute. Hold on. But it's just a chance for you to see the questions and get some understanding of what's going to be expected of you when you take the real one the following Monday. Again, you'll be taking both the lab and lecture exam on Monday the 14th. That is the only thing we are doing on Monday the 14th. There is no lecture on that day. So you have the entire class time to take the exams in either order that you want. So if you want to take the lab exam first or the lecture exam first, that's up to you. However, both exams must be completed during class time. So they will not be available till 8, and they shut down at 12:35. And again, like the quiz, again, remember that doesn't mean that you have till 12:35 to is when they end. Each exam is going to have a time for it. So when you start the lab exam, you will have 80 minutes or whatever it is to be able to complete it. So 80 minutes after you start is when it's going to be due. However, the time can't go past 12:35. So if you start the lab exam at 12:15, you are going to have 20 minutes to complete that exam, because the exam must be completed within the class time. So again, and all of that information is on there. There's a running clock to tell you what it is and what things are going on. So hopefully that makes sense. As far as the labsters, you do not need to continue to get a perfect score. You're welcome to do that if you like. As I mentioned, remember, for full credit, you must get it at least 80% correct or more. Now, again, this is synced with Canvas. So when you complete that activity, it puts a prorated score into your grade book automatically. However, as long as you complete 80%, I go back, and actually that's what I was doing while you guys were taking the quiz. So if you go and look now, everybody except one person has completed it with 80% correct. So I went and changed those grades to full credit. So you do have full credit. If, again, for the learning experience, if you want to go back and get a perfect score, you're welcome to do that, but it is not required. Lab and lecture exams, they're not split in half evenly. Again, it depends on the length of it. This lab exam probably is going to have, I'm not finished working on it yet. My guess is somewhere around 70 questions would be my guess of where this one is going to be, which means you'll probably have usually I do about 15 minutes more for the lab exam. So you'll probably have about 85 minutes to be able to complete that. The lecture exams, depending on how many essay questions there are usually between 2 and 2.5 hours. So it's 2.5 hours, for that, figure an hour and a half for the lab exam. So that's about 4 hours right there worth of tests. And again, that's the time that it has available to you. Most people don't use the full 2 or 2.5 hours on the lecture exams. I don't know how the total, my guess is somewhere around, 70 questions on the lab exam. So So lab exam will probably have 70 identification, yeah, somewhere around there, 76, somewhere between 60 and 75 would be my guess, somewhere within that range. Probably not 60, probably 65 to 75, somewhere in that range is how many questions your lab exam will have. The lecture exam is going to typically have somewhere around 30 multiple choice questions, then somewhere around 2 to 6 fill in the blank questions, and then usually somewhere around 8 essay questions. No, Christopher, hold on, I'll get to that. And that's a great question. So yes, to answer your question, Christopher, unfortunately, despite my best efforts, I did not scare anybody off since Wednesday. So unfortunately, I do still indeed have a full roster. So I am unfortunately not going to be able to add anybody else. So anybody who is still on the wait list, unfortunately, I will not be able to add you today. And at this point, if I'm not able to add you today, I'm just not comfortable adding anybody after that. Again, if you're on the wait list, I don't expect you've purchased the resources and have been able to maximize your efficiency on this, and you just keep falling further and further behind. And with the exam on the 14th coming up very, very rapidly, I will not be adding any more students this semester. So I do apologize to anybody who is on the wait list. I will not be able to add you. Like I said, I tried to be super scary last week, but apparently it didn't work because everybody is still here. All right. Good luck. I thank you, Chris, for coming. I appreciate that, and I apologize to anybody else as well. Okay. Well, so apparently some people are terrified, but they're not terrified enough to drop. All righty. Excellent. So that's the game plan. Any questions on any of that? Laura's question, which one was that? Did I not answer that? Oh, okay. Is it going to be normal for the daily quiz to be a topic? So again, as I said numerous times last time, and I mentioned before, typically coming in on Monday, it's not uncommon to have the information, or coming over vacation, it's not uncommon to have the information highlight some of the things we're going to be talking about, because if you've had 5 days off, I expect you had to have been starting to look at the material that you are going to be responsible for. If we have a quiz typically on a Wednesday, then typically that will be the material that we covered on Monday. And again, that's not always going to be the case, but it isn't a bad idea coming in off of a holiday or coming in off of a Monday to make sure that you've at least looked a little bit on the information that's coming ahead. Obviously, I don't expect you to know everything about it, because then there wouldn't be any point in me lecturing. But some of the basic concepts that are involved in it, I think are reasonable to ask. And that's what these quizzes are about. Just an encouragement to remind you to be thinking about the material that we are covering in this class. All right. I think I only partially answered the question. So again, you are correct. You have 4 hours and 35 minutes to complete the exams. Most people, but the two exams combined are going to be somewhere around 4 hours. So typically what I encourage students to do is to do the lab exam first. Most people who have technical issues, it is because of the lab exams where there are 70 pictures that need to load into Canvas to be able to load into your proctorio quiz for you to be able to identify them. So most of the time when people have questions or problems, those problems relate to that lab exam. So I encourage you to start that early. Then once you get that completed, your test taking style. Do you like to take a long time with your essay questions and really think about it and cogitate on them? Or do you like to rush through them? So again, feel free to take a break. In fact, I encourage you strongly to take a break in between the two exams to give you a chance to, get something to eat, get something to drink, go to the bathroom, cry, whatever it is you feel you need to do during that time. But just make sure you pay attention to the time. It is better to start early and finish early than to think that it's only going to take you an hour and a half to answer the essay questions. And so you start an hour and a half, you start at 11, and then you end up needing more time than that. So it's better to make sure that you have the time, because at 12:35, like I said, whether you've been in it for 2 hours and 29 minutes or whether you've been in it for 3 minutes, it shuts down at 12:35. So it is better to start early rather than late, especially in the first exam while you're trying to figure out how this whole process works. All righty. Excellent. Any questions on anything else then? All right, excellent. With our game plan under our belt, and look at that, we finished it in far under 30 minutes, which is what it usually takes us with all the introduction. See, once we start getting the hang of this, we can get more and more quickly into the material. And so let's do that. Let's get into the material.
Can I say one thing real quick?
Yeah,
Um,
absolutely.
you know what? You're messaging me privately, not the class. So switch it back to everyone.
Oh, I'm sorry.
I'm asking questions, but it's just going to me, so I want to make sure he knew.
Oh, I apologize for that. What have I said to you that was, did I say anything to you that was important that I needed to say to everybody else? From me.
Let's see. He said that there might be 8 or more essay questions.
Well, I didn't type that, did I? I thought I said that out loud.
No,
Oh,
it's
this is,
the classmate that's messaging me privately. I just wanted to tell him because I was trying to
oh, there's a
tell him in
student
the
that is.
chat, but he's
Okay,
not reading it. So
got it. All right. Sorry. That is, all right, excellent. So are there any other questions or anything that anybody had that I need to answer? Maybe they're just shy and they wanted you to ask the question for me. Yeah. So I tell people to do in class, just pay the person $1 next to you in class and have them ask the question. All righty.
Actually, I have a question.
Yes.
So, like, if we get stuck on some questions and it's past Friday where we can't get help from Jeff, is there any way possible that you could like return the question or return the answer like pretty quickly?
I'm not sure I'm understanding your question. So what are you asking?
Is there another way that you can respond like quickly to questions that in case there are some questions that I get stuck on?
Yes. Obviously, office hours are one of the best ways, like especially because they're right after class and stuff like that, for us to be able to talk about materials, because we also can interact back and forth. But I'm very good at responding to emails. If you have questions, the best thing to do is to send me an email. And usually I'm pretty good at responding to emails. It takes me at worst about 24 hours, but usually I'm able to respond to emails much quicker than that. Obviously, it depends on what's going on. And then I would also say typically, especially the weekend before an exam, I try to make an extra effort to be more responsive to emails, just because I know the timing is an issue of that. I mean, obviously, it depends on the information and the time. If we have we have an 8 o'clock in the class morning on, I mean, a question. Try that again in English. We have an 8 o'clock in the morning class on Monday. So if someone's up late doing homework at 10, 11 o'clock at night and sends me an email there, I'm not likely to get that because I'm probably in bed or already getting ready for bed at that period of time. But I do try to be very responsive to email. So I think. I think that's usually the best way to get a hold of me that way. Okay.
So I have a question, even though it's private, not related to all class. And
Okay.
my question is about having access or getting access to the code, like to the book code. Can I like purchase only the code, not the book? Like, can I do that?
Yes. So yes, the Master in A&P can be accessed. So when you log on to the Master in A&P, you do have the option to purchase it without the electronic version of the textbook. And I think without the textbook, I think it's like $66, $70, something like that. And then with the textbook, it's like $115 or $116 or something along those lines. So yes, you can get access to all of the things on the Master in A&P.
Thank you, sir.
Yep. Allison, no, I don't repost the chemistry quiz because I use it again and again. So I don't make those questions available. If you have a question about it, then the best thing to do is come to my office hours and we can bring it up and go through it together that way. But I do not post the answer. So if you're curious about what you got wrong or anything like that, I'm happy to go through that with you on my office hours, but I do not make that test available because I reuse those questions. All right. Any other questions? All right, excellent. On to cells. Yes, everything we've talked about in this test. So the stuff that you covered on the chemistry quiz, like I said, aren't my questions because that's stuff that you should have known coming into this. But the whole lecture we spent in the last class, half of it was on chemistry, was on the macromolecules, was on, lipids and carbohydrates and proteins and nucleic acids and all that. And is that a part of what we talked about in class and lecture? Is that something that's going to be on the exam? Absolutely. All right. All righty. I'll pause a little longer this time because every time I think we can get started, someone asks us a great question. So I'll pause a little bit longer. Any other questions? All right, let's talk cells. So again, one of the things that we talked about when we were doing our introduction is one of the big difference between the chemical level and the cellular level, aside from an increase in complexity, is that when we talk about cells, cells are the smallest structure that we consider to being alive, right? And there are plenty of single-celled organisms. In fact, single-celled organisms rule the planet. There are far more single-celled organisms on this planet than there are multi-celled organisms. In fact, they did a study once where they talked about if they were to take you, if you were standing there in front of your loved ones and they removed all of the cells that were you, people would still see something standing there because there are so many single-celled organisms both inside and outside your body, covering the surface of your body, internal inside of your body, and things like that, that basically you would still see something that looked like a person or a person-shaped bundle of trillions of single cells just sitting there. You know, a single square centimeter of your skin has over 10,000 bacteria on it. I know when I say that, especially these days, everybody goes diving for their Purell. But as we've talked about, those are a good thing. Those are resident bacteria, and they actually play an important role in your body's protection. However, to become the complex multi-celled organisms we have, cells have to become specialized, right? And there's both good and bad to that. If I have a single-celled organism, where are my drawing tools? If I have a single-celled organism and I take that single-celled organism and I cut it in half, what happens? It's the part where you answer. Not a trick question. Take a single-celled organism, chop it in half, what happens?
It becomes two single-celled organisms?
Exactly. It becomes two single-celled organisms. They both run off and live happy lives. Right? Same thing happened to you. I cut off your left hand. Does your left hand run off and have a happy life? No, exactly. And the reason for that, the difference for that, the reason your hand can't run off and live a happy life is to become multi-celled organisms, our cells have to become specialized. That single-cell organism is capable of doing everything that is necessary for life. Responding to an environment, metabolism, transportation, reproduction, growth, all of those things that we talked about that are characteristics of life, this single-celled organism is capable to do. However, to become a multi-celled organism, some of these cells in our body become highly specialized to do something that helps us as an organism as a whole, but that means they aren't necessarily able to do the things that are necessary to do, right? To be these big, huge, complex monster that we are, there are cells that are inches away from the surface of your body. And if you have a cell deep inside of the body, is it able to get the oxygen that it needs from the environment? Can we rely just simply on diffusion to get cells all the way to the core of our body?
No.
No, we use our cardiovascular system to get the oxygen that it needs to those cells instead. And so while they become specialized, it allows us to become big and complex, but at the same time, those cells become interdependent on each other as well. And we have lots of different types. How many different types of specialized cells do you think we have? Well, I just spoiled it, but tell me. That should make it easier to answer the question.
A lot.
A lot, absolutely. Although eventually we need to get, there you go, over 200. And that's just in humans, right? You can start counting them out. We got eyeball cells and toenail cells and liver cells and so on and so forth as we go through all of those, right? Other animals have different types of cells that we don't have. And don't even get me started on plants. I mean, who knows what those weird things are, right? There's all sorts of different types of cells. Here we have a pretty picture from your textbook that shows some of the dramatic diversity of cells we have. We have cells that are specialized for movement. We have cells that can actually propel themselves. The spermatozoa is special in that it is able to propel itself. Some of the nerve cells in your body can be over 1 meter long, right? The cell body starts in the small of your back and goes all the way down to your big toe, right? If it's someone like LeBron James, that's like 85 feet, right? These cells can be massive. We have cells highly specialized for transportation, like our blood cells, epithelial cells for movement of material inside and outside of our body. We have all this tremendous specialization. None of these cells really look like each other, but there are some key components that every cell has, and that's really what we're going to focus on. We're going to focus on what a typical cell is comprised of, and its anatomy, its basic anatomy that allows it to have its basic function. But obviously, as we work our way through 430 and 431, we'll run into lots of examples of specialized cells that have specialized functions. Now, with over 200 different types of cells in your body, those 200 different types of cells in your body can all be divided up into one of two main categories. What are the two main types or categories?
Sex cells.
There you go, absolutely. One of them is sex cells. And what's the other one?
Somatic cells.
Somatic cells, excellent. What does somatic mean? Anyone know what the root of soma means?
Soma mean body.
Body, exactly. So the somatic cells are basically what make your body. And what do sex cells do?
Sex
For reproduction.
Yeah, if you think about it, they make someone else's body. All right, so somatic cells make your body, sex cells make somebody else's body. Those are basically the two main types of cells that we have in our body. Now, again, cells are important because they're the smallest unit capable of performing all of the vital functions, physiological functions, right? This is what allows us to define them as alive, right? Because they're able to do all of these things. To be able to be successful, each cell must be able to maintain its own homeostasis. But the important thing to remember is that all cells come from the division of pre-existing cells. All right? Started the simplest case. How many cells did you start as? 1, right. What do we call? There's a special name we call that cell. I mean, you could kind of argue 2, because you have the egg and you have the sperm that are going to come together. Oops, if I spell it right. But that egg and that sperm, when they come together, they form a special cell. There we go. That's the magic word I was looking for. That
zygote.
Absolutely, that zygote. They form a zygote. And then that one zygote became 2 cells, and those 2 became 4, and those 4 became 8. And then suddenly you have the trillion cells that are in your body right now. All right? We have a special name for cells that divide rapidly to produce other cells. What do we call those? What do we call cells that make other cells? There you go. We call them stem cells. Right? Notice someone had to type it. No one was willing to say it out loud because stem cells is almost like a bad word, right? Someone says stem cells, and you've got to go get pitchforks and torches, and right, we have to get a mob together because of that. But again, I think there is some misunderstanding on exactly what stem cells are. So I think it is important that we define what a stem cell is. A stem cell basically has 2 main criteria to it. Oops. The first is that they are undifferentiated. Oops, if I spell that right. What does it mean to be undifferentiated? Anyone know what it means to be undifferentiated?
Not to be separated. Or
Sort of, although actually I like what Haley said. Doesn't have a specific function, right? It really has no specific function. It's not been specialized yet. It doesn't have a job. It's basically a millennial, right? Undifferentiated cells aren't specialized, don't have a function. Say it this way. But stem cells do one thing. And that one thing that stem cells do is these cells are, and let's say it first in the fancy way, they're highly mitotic. Ugh. But again, that's just a fancy way of saying that these are cells that divide rapidly. Right? Or like Haley's mentioned, they reproduce fast. Excellent. That's it. Period. End of paragraph. That is what a stem cell is. Now, the key is that there are three main types of stem cells.
Professor, did you mean to say that they are not specialized for
Oh, yes.
differentiated?
Ah, thank you. Thank you for catching that. You are absolutely correct. And unfortunately, I can't go back and change that. So let's cheat and do it this way. Thank you for catching that. Thank you very much. There we go. Excellent. Yeah. Talk about changing the meaning. Absolutely. Excellent. There you go. So there are three main types of stem cells. The first type of stem cells are what are called unipotent stem cells. What do you think the definition of a unipotent stem cell is? Uni means one. Excellent. So in this case, unipotent stem cells are, again, cells that are undifferentiated, cells that are highly mitotic, and when they divide, they produce only one type of cell. Now, I know you probably haven't thought of it in this way, but I know you're absolutely positively aware of it. Because tell me one of the things you know about your skin. What do you know about your skin?
Elasticity.
Okay, it has elasticity. What else do you know about your skin?
Protective,
It's protective. What's one of the things that makes it protective?
a barrier from the outside to the inside.
True. How does it form a barrier? What do you know about the topmost layer of cells of your skin?
It's multi-layered and the outside layer dies off.
Is dead. Absolutely. It's dead, right? Constantly sloughing off, right? As you look around the table in front of you, the dust that is on your computer screen, the dust that is on the table or the kitchen table, the vast majority of that are your skin cells and the skin cells of other people and animals in your house, right? We are constantly losing millions of new skin cells, losing millions of old skin cells every single day. So why aren't we down to muscled here? Because we also produce millions of new skin cells, right? You have tiny little stem cells in here that divide to produce millions of new skin cells every single day. And while those cells divide to produce millions of new cells, how many different things can those new cells become? One. Skin cells. That's it. All right. These are different from pluripotent stem cells. What's the definition of a pluripotent stem cell?
Many.
Right, excellent. When it divides, they have to be consistent. Produce. I didn't sleep well last night. If you can't tell, I get the alphabet soup speak when I don't get a lot of sleep. When it divides, they produce many different types of cells. Excellent. An example of this is in your bone marrow. In your bone marrow, you have a cell called a hemocytoblast. You don't have to write that term down yet. We'll get to it eventually. But that hemocytoblast is a cell that is capable of dividing to become any of the cells associated with our blood. That means red blood cells. That means any of the 5 types of white blood cells, and even the cells that make our platelets. Platelets are really just cell pieces of another cell that is ripped off. So any cell associated with the blood can be made by these pluripotent stem cells found in the red bone marrow. Now, while it can form many kinds of cells, is that hemocytoblast in our bone marrow capable of making skin cells? No.
No.
So pluripotent cells can become many different types of cells, but they can't become all types of cells. Are there stem cells that can become all types of cells? Yeah. Those
Multiple.
are omnipotent
Omnipotent.
stem cells. All right? You and I and everybody else standing around you have millions of unipotent and pluripotent stem cells inside of you, capable of producing all sorts of different types of cells in your body. But what you and me and the person standing next to you don't have inside of them, I guess unless you're pregnant, is omnipotent stem cells. Omnipotent stem cells, when they divide, they can produce any type of cell. These omnipotent stem cells are only found in like the zygote, in embryos and fetuses. Basically, by the time we're born, those omnipotent stem cells are gone, right? This is what's so frustrating about this process. We don't have these omnipotent stem cells. So let's say, for instance, you played Superman in several really good movies and one really bad one. And then one day you're out riding on your horse and you fall off the back of your horse and damage your spinal cord. You spend the rest of your life in a wheelchair because we don't have the means of being able to replace those nervous cells that have been damaged, right? Omnipotent stem cell is a really interesting field of study where we have these cells that could potentially become anything. So theoretically, right, if you have that spinal cord injury, maybe one day it'd be possible to take omnipotent stem cells, put them in your into your small of your back, have them divide and become nerve cells and replace those that have been damaged. The potential for the use of these is amazing. Or maybe we could, grow you a new heart if you're having heart problems, or a new liver if you're having liver problems. The problem with it is, where do you have to go to get these omnipotent stem cells? Embryos and fetuses, right? And when you have to go to that embryo and fetus, then there's all sorts of religious, moral, social connotations, ethical reasons for it. Absolutely, right? So there's all these issues from it. Now, one thing that I would say is one of the sources from it that doesn't have the same ethical type of connotations is umbilical cord blood. When people, yeah, exactly. I was just going to say that, Haley. Thank you. When people give birth, one of the things they have the option of doing is collecting the umbilical cord blood. Some people actually save it and store it, freeze it, so that there are some omnipotent stem cells for that particular baby, so that if something happens down the line, when they, maybe they decide to drink vodka for breakfast, lunch, and dinner for 17 straight days as an adult, you have the ability maybe by then to be able to grow them a new liver, or it can also be donated for research as well. So there are less ethically charged ways to get them, but there aren't as many of them, and so
It's expensive too.
Yeah, it can be expensive. Yes, storing them is absolutely expensive. Obviously, it doesn't cost you anything to donate it, but yeah, if you want it for personal reasons to store it and keep it, then yes, it is definitely expensive. But again, because of the social, ethical, all those religious connotations of those, that's why when people hear stem cells, right, they think dead babies and they want to grab their pitchforks and their torches. but that's not what we're talking about. So I want to make sure you understand that there are different types of stem cells. Every single one of you has stem cells inside of your body right now, right? So again, that's not what we're talking about. All right. Questions on that? Bit of an aside, but I think it's a very important insight that we understand what a stem cell is and that there are different types. All right, excellent. We have perfect understanding of that, which no questions tells me that is the case, then we will move on. Excellent. When we talk about a cell, there are basically three basic components to it, right? Normally, if I ask you to draw a cell, you're going to, draw a big circle, and inside that big circle, you're going to draw a little circle, right? Something like that. And this is the basics of what it is, but there are basically three things that we really see here. The first is the nucleus. That's the littler circle that we saw on the inside. The second component out here is our plasma membrane. And then basically the third component is everything else in between. And everything else in between the plasma membrane and the nucleus is called the cytoplasm. So those are our three components: the nucleus, the plasma membrane, and the cytoplasm. So we have our nucleus, which, as we've talked about, contains our DNA, our cytoplasm, and our plasma membrane. And if you didn't like my amazing drawing, here is another one showing you a better sense of all the hunk and junk associated with all of these things. All righty, let's start first by talking about the nucleus. When we talk about the nucleus, most cells have one. What is the key word in that sentence?
Most.
Most, because what does most mean?
Many, but not all.
Yeah, not all, exactly. And that is the key. It means not all. So there are some cells that actually have multiple nuclei. So there are some that have multiple. And there are also some cells that, at different stages of their lives, have no nucleus. Can anyone give me an example of a cell that at a certain stage in its life doesn't have a nucleus? Yeah, red blood cells, absolutely. Obviously, red blood cells have to start with a nucleus, but then they basically fill themselves up with hemoglobin so much that they're all, you know what, I don't need this pesky nucleus anymore, and they chuck it. And they no longer have a nucleus. Now, once they chuck their nucleus, are they going to be able to survive for very long? No, and in fact, a red blood cell only lasts about 100 to 120 days. But during that 100 to 120 days, it is a cell without a nucleus. Excellent. Now, when we talk about the nucleus is a membrane-bound organelle. Organelles are clusters of chemicals and other components inside of a cell that serve a specific function. What do you think it means to be? It means to be membrane bound. Yeah, it has a membrane on the outer surface of it that surrounds it. Now, when we were talking about macromolecules, when we talked about plasma membrane, what macromolecule did we say was responsible primarily for producing those plasma membranes?
Lipids.
Lipids. And does anybody remember which specific lipid?
Bilayer phospholipid.
You're right. It is. There you go. It is a bilayer of phospholipids. Excellent. So a bilayer of phospholipids. So again, if I draw a nucleus, right, as that circle, it is comprised of a bilayer of phospholipids. But actually, what's interesting about the plasma membrane of the nucleus, as you see from the picture, is it is not one, but two membranes. Or they have a fancy name for this. They say it has a double membrane. So actually, what it actually has, as you can see from the pretty picture, is not one, but two. Stop it. There we go. Plasma membranes. And so if you think about it, there are really 4 layers of phospholipids. 2 layers of phospholipids in each one of those membranes, and 2 membranes total. So it is a double membrane, 4 layers of phospholipids total. So a total of 4 layers of phospholipids. Excellent. This fancy structure that I've drawn has a name, and that name is the nuclear envelope. The job of the nuclear envelope is to protect the DNA that is located inside of it. But as we've talked about, we have this pesky issue where the DNA is the information for making proteins, but all the hunk and junk that actually does the making of the proteins is outside the nucleus. So while we want this nuclear envelope to be big and protective, we do need a way to get information into and out of it. So notice as you look at the pretty picture here of the nucleus, there are these little individual, and I'll just draw them as a solid line here, structures, these pores, spaces really. So notice this is a structure. Vocabulary is important. This is a space that is formed. And this space that is a passageway through the nuclear envelope is what is known as the nuclear pore. So there are all these nuclear pores on the surface that are these openings. It's actually, maybe let's do it this way. This is actually probably a better way to do it. There we go. So there are these pores in our nuclear envelope where materials can get in and out. Not the space between the phospholipids, the passageways between the 2 layers. There is a name, quite frankly, there is a name for the space in between, but you don't need to know what it is. Through the nuclear envelope, into and out. So again, if you look at this illustration, the purple represents the nuclear envelope, and these orange structures are forming the spaces, the holes in it, that are the nuclear pores. And that allows information to get into and out of the nucleus. So materials, information, resources are able to pass through these spaces, through these nuclear pores, to get inside and outside of the nucleus. All right. Questions on that? All right. Actually, let's not do all of that. I just want to get rid of this. Bing, boom, boom, pop, beep, boop. Perfect. All right. Now, actually, let's cheat. I'm going to go back. There we go. Now, as I mentioned, and I'm going to cheat again and draw my nucleus. Inside of the nucleus, as we mentioned, is our DNA. But DNA needs to be protected. Not only does it need to be protected by being in the nuclear envelope, but it can't be here in the nucleus by itself. So what happens is DNA has lots of proteins that assist it and protect it. So when we look inside of our nucleus, we see a big cloud of material. And that cloud of material is not just DNA by itself. Instead, what it is a combination of DNA and proteins. Now, we could just keep saying the stuff inside the nucleus is a combination of DNA and proteins and say that every single time, but that gets exhausting to have to say that every single time. So they give it a fancy name. The fancy name that they give for it is chromatin. Chromatin is the loosely, let's say it this way, the loose DNA and protein. It's like a big bowl of spaghetti. And that allows us to be able to access the DNA to do work. So that absolutely positively is a good thing, that we have all the DNA and proteins loose within the nucleus, this loose bundle of chromatin, so that it allows us to access it to do work. But as we know, all cells come from pre-existing cells. So eventually this cell is going to need to divide. And how many people here have eaten spaghetti before? A couple of you. Excellent. How many people have shared a bowl of spaghetti with somebody else before? Less of you. Okay, that's all right. Some of you don't want to share. That's all right. But let's theoretically say you were going to. Let's say you had a big bowl of spaghetti you wanted to share with somebody else. Would it be really easy to separate that bowl into half? Yeah, sure. You just use a knife, cut it in half, and slide it that way. But what if you had to separate it without breaking any of the strands? Would it be easy to divide a bowl of spaghetti into two without breaking any of the strands?
No.
No.
Absolutely not. Well, unfortunately, that's what we have to do when this cell divides. So, aside from protecting the DNA, the other thing that the DNA helps, the other thing that the protein helps the DNA to do is when it comes time for the cell to divide, the proteins help the DNA fold up into a tightly packed structure. And so while our chromatin is the loose DNA and proteins, when it is time to divide, the DNA and proteins tightly bind up into a structure we call a chromosome. So a chromosome and chromatin are exactly the same stuff. The only difference between chromatin and chromosome is the shape. All right, so again, let me erase this. So again, we have the loose chromatin, allows it to do work, and we have them bundled up into a tightly packed chromosome. Again, this is when the cell is doing cell business. When the cell is working, that's when we have our chromatin. When the cell divides, and only when the cell divides does it bind up to become chromosomes. Now, remind me again, what does the DNA contain? What type of information does the DNA contain? What information does DNA contain? It's the genetic code, true, genetic info. But what does it tell us what to do, how to do? What do we do with that information? There we go.
Protein.
DNA is used to make proteins. It is the instructions to make proteins. So when it is loose chromatin, if we want to make a protein, and we will actually describe the process in this class, we're going to do it on, we're going to do it on Wednesday the 9th. We will actually talk about and learn the process by which we make a protein. When it comes time to make a protein, we need to find the little piece of DNA that has the correct information of how to make that protein. We need to make a copy of it and get it ready and do all these things. And it requires a lot of hunk and junk to do that. A massive number of proteins, a massive number of enzymes, a massive number of nucleic acids, all this stuff. So what happens is, and let's kind of draw a simplistic illustration of it. Let's say here is a little bit of one of the strands of DNA. Well, all those proteins, all those enzymes, all those nucleic acids, they're all going to clump together around this one single location where where it has the instructions to make that protein. Now, the chromatin is very, very tiny. we can't see it under the microscope. When you look at a microscope and the cell is just doing its work, the nucleus just looks like it's a cloud of fuzzy material inside. When it folds up into a chromosome, those chromosomes are so tightly packed that we're actually able to see the individual chromosomes. Anybody know how many chromosomes we actually have as humans?
23 pairs.
There you go, 46 or 23 pairs, right? Pairs, because you got one from mom and one from dad. So 23 pairs, 46 total. Absolutely. And those can actually be seen with a light microscope when you're looking at them. The chromatin can't. However, all this hunk and junk that wraps around it becomes so big and dense, it actually can be seen with a light microscope. And that's what this right here represents. This big blue ball in the center of our nucleus is all of that material condensed around the DNA. Basically, it represents where protein synthesis is starting. And we give a fancy name for that structure, and that fancy name for the structure is the nucleolus. So nucleolus is a dark spot in the nucleus where protein synthesis is starting. Now, anybody know about how many genes we have in our genetic material? Too many, yes. Tens of thousands of them, absolutely. Which means we can make tens of thousands of proteins. And do you think the cell needs to just make one protein at a time? And then it just has a massive queue of everybody waiting in line for their particular protein to be able to be made? No, it can make many proteins at the same time. Of course, each time it did that, we would need another cluster of material to occur within here, and then a third or even a fourth or a fifth. So nucleolus is the singular, whereas the plural is nucleoli. So nucleoli is the plural, nucleolus is the singular. You have one nucleolus, you can have 3 nucleoli. All right.
You can have numerous nucleoli within each nucleus.
Yes. So yes, if theoretically, if you were a cell that had multiple nuclei, each nucleus would possibly have one or more nucleoli. That is correct. Excellent. So there you go. We now know what a nuclear envelope, nuclear pore, nucleolus, nucleoli, chromatin, and chromosomes. Fun with vocabulary. Like I said, learning biology, anatomy and physiology in particular, is like learning a new language.
Can you say that one more time? The nucleoli, plural, is within cells with more than one nucleus?
No. If you have one nucleus, that nucleus can have more than one nucleolus in it. So again, remember, a nucleoli is just a cluster. So you can, so the way I interpreted the question, so let's say it this way. Fun with pictures. You have a cell, and that cell has a nucleus. Inside of that nucleus, you could have 2 nucleoli. Okay? So nucleoli, 2 nucleoli within a single nucleus within a cell. Remember, we did mention that some cells can have multiple nuclei. If a cell has multiple nuclei, inside each nuclei, you could have nucleoli. So you could have many nucleoli inside of the nuclei. And again, I did 2 in each of them. It doesn't have to be 2 in each of them. It could be 1 in one and 5 in the other. But yes, if a cell has multiple nuclei, multiple nuclei, it can have multiple nucleoli in there as well. Fun with vocabulary. All right, but you don't have to have multiple nuclei to have, you don't have to have multiple nuclei to have multiple nucleoli. Again, fun with vocabulary. No, remember the nucleoli is not storage. It's not storing information, it is reading the information in the DNA. The DNA is the instructions to make a protein. And again, we'll see how this process actually works, how we make a protein. And it starts by reading the DNA in the nucleus. So when I'm making a protein, that making a protein starts by clumping a bunch of stuff around the DNA inside of the nucleus. And that big clump of stuff around the DNA in the nucleus is something that can be seen with a light microscope. And so they gave it a name. It's a nucleolus. But yeah, no, it's not storing anything. It's not storing the information. It's not there all the time. It's there when we're making proteins. If we're not making proteins, there's not going to be a nucleoli. All right. Questions on that? All right, let's take a closer look at this chromatin. Notice here we see our DNA, and as we briefly mentioned, DNA is in a double helix, 2 strands of nucleic acids bound together, and we'll talk about the rules of how that works, although you guys already mentioned some of that the last time. And here we see an example of how some of the proteins can work with it. Notice in particular, these proteins are special globular proteins called histones. And notice what these histones do is help to support the DNA. So notice the DNA is wrapping around these histones in that loose chromatin. And again, when it's time, these histones and other proteins help the DNA to condense down into this tightly packed chromosome. So again, when it's a loose bowl of spaghetti, it's chromatin, DNA, and proteins. And then when it's packed up tight, ready to be transported and moved around, it is a chromosome. All right. And like we said, we have a total of 46 chromosomes in each one of our somatic cells. All right. Like I said, we'll talk more about the nucleus and the DNA when we talk about actually mitosis later on today. We'll talk about that. But what we need to talk about now is the cytoplasm. There are 3 main components to the cytoplasm. The first is the water, the cytosol, our intracellular fluid. Remember, we talked about intracellular fluid is where most of the fluid is found inside the body, inside of the cells. Of course, it isn't just, and let's draw another cell for this. It isn't just water by itself. There is all sorts of other hunk and junk in there as well. Things like ATP, things like amino acids, things like sugar, things along those lines. But it makes up about 55% of the volume of the cell. And it is mostly water with a few other things added in, like amino acids and lipids and all sorts of fun stuff like that. But one of the other really, really important things we find inside the cytosol are ions, positively and negatively charged ions. And what's important about these ions, actually, let me go ahead and erase that for a second. What's important about these ions is that there is an unequal distribution of ions inside and outside of the cell. This is important because it allows the cell to do work. A lot of the work that a cell is able to do, it is able to do because of this unequal distribution of ions inside and outside of the cell. Now, just out of curiosity, who here knows what the most common cation is in the body? Let's start easy. What is a cation? The opposite of a dog ion?
Potash.
Say again?
Potassium?
Absolutely. So, so again, positively charged. Excellent. A cation is a positively charged ion. What is the most common positively charged ion found in the body? Not in the cell, but in the body. Be careful.
Potassium?
Calcium.
So you guys, I get where you're going with this. The most common cation inside of your body is sodium. All right, sodium is that. However, I think one of the reasons this question is a little confusing is there a lot of sodium inside of the cells? No. Most sodium is located outside of the cells. So there's a massive amount of sodium outside of the cells. And inside of the cells, oops, no. Inside of the cells, there is a very small amount of sodium. So sodium is the most common cation in the body, not the most common cation inside the cell, as some of you guys pointed out. Absolutely. The most common cation inside of a cell, intracellular cation, is indeed potassium. So there's a massive amount of potassium inside of cells and very little potassium outside of cells. What's another important cation that is unequally distributed inside and outside of cells?
Calcium?
Calcium, absolutely. Calcium makes cells do wonky things. If you are a cell and you want to do a wonky thing, like, oh, I don't know, produce an action potential, or if you're a muscle cell that wants to contract, or you're a gland that wants to release a substance, pretty much calcium is what helps you to do that. So not surprisingly, there is a tiny amount of calcium inside the cell, because we don't want to be doing wonky things all the time, do we? No, absolutely not. So we have a tiny amount of calcium inside the cell, but lots of calcium outside the cell. Excellent. We've done a really good job of identifying all of the important cations. And again, there are plenty more, magnesium and all sorts of other fun things, but these are a good three that we'll spend a lot of time talking about. But notice we haven't mentioned any anions yet. What's the most common anion? What's the most common negatively charged ion? Come on, I know you guys know.
Oxygen.
Chloride, there you go, absolutely. And where do you think there's more chloride? Inside the cell or outside the cell? Outside, absolutely, right? We're big bags of salt water, basically, right? Huge amount of sodium, huge amount of chloride. Outside of the cell, put those together, you get table salt. We're all big bags of salty water, and very little chloride ions inside of the cell. Excellent. So here are probably our four most common and the four ions we will talk the most about inside of our cell, but here we get a sense of them. And again, there is this unequal distribution. Large amount of potassium inside the cell, large amount of sodium, chloride, and calcium outside the cell. And this unequal distribution is one of the important things that allows us to do work. All right, we got that. I need to erase this. So any questions on this before I erase it? All right, excellent. So like I said, we have an unequal distribution of ions, like high levels of potassium inside the cell and low levels of sodium inside the cell. The second component of our cytoplasm are what are known as inclusions, right? Every cell has ATP inside of it. Every cell has potassium and sodium inside of it. Different levels, but they all have them. However, there are some specialized chemicals that are only found in one or a couple of cells, right? A great example of this is one of the ones we already talked about, hemoglobin. Hemoglobin is a special transporting protein. What did it transport again?
Oxygen.
Oxygen.
Oxygen, absolutely. Right, used to transport oxygen. And really, it's only found in one cell. What's the one cell that contains the special inclusion hemoglobin?
Red blood cells.
Red blood cells, absolutely. Right, others may have massive amounts of glycogen. Remember, glycogen is our storage molecule for glucose and things along those lines. So inclusions are special molecules that are found in one or just a few cells. It varies by cell. Your book's got a list of them, but the list is exhaustive. So what will happen more likely is, as we talk about cells, we'll talk about the special things we find inside of them. We'll talk about their inclusions, but it's important to know that they're there. And the other thing you find inside of cells are organelles. Organelles, just like you have organs inside of your body, organelles are the structures inside of a cell that help it to serve its functions. And organelles come in two flavors, membranous and non-membranous. What do you think the difference between a membranous organelle and a non-membranous organelle is? Yeah, they have a membrane, absolutely. Or membranous organelles are bound by a membrane, and non-membranous organelles are not bound by a membrane. There you go. And it's just that simple. All right. So what we are going to do, looking at the time, is this is a good stopping point. We will go ahead and take our first break here. And when we come back, we will start discussing our organelles. So any questions before we take our break? All right, looks like it's 9:17. Let's take a 15-minute break. So that means coming back at 9, what is that, 32? At 9:32, we will restart, and I will start the recording at that time. All right, any questions? All right, see you in 15 minutes. All righty. Any questions before we dive back in? All right.
I couldn't see the bottom of the organelles number 3. It's kind of large.
Oh, you mean what it says underneath the organelles?
Yeah, I know
Okay.
it says none, not bound and bound.
Oh, what I had written. Oh, okay, I'm
Yes.
sorry. Yes, basically membranous means that it is bound in a membrane, and non-membranous means that it is not bound in a membrane. And obviously we're going to talk about those in more detail now, but like I said, hopefully those definitions are pretty intuitive, but great. I'm sorry. I appreciate that. I know different people viewing this different ways don't always see all the bottom, so I try to keep as much stuff up as possible, so I apologize for that.
No worries. Thank you.
All righty. So let's start working our way through some of the organelles. We're going to start first by talking about the non-membranous organelles. And again, remember, as we just mentioned, that means they are not bound by a membrane. And the first of these is an organelle known as the centrosome. The centrosome is a dense area of the cytoplasm, but it is not contained within a plasma membrane. The centrosome is also sometimes referred to as the microtubule organizing center. So what is it that you think the centrosomes may do? Well, if their other name is the microtubule organizing center, what do you think centrosomes do? Not a trick question. Says it on the board. Organize. Excellent. They organize something. What do you think they organize? Microtubules. There you go. All right. These are the easy questions, folks. Absolutely. It organizes microtubules. Now, of course, you may not know what a microtubule is, and we will actually talk about that a little bit later. They're an important part of the cytoskeleton. But in particular, what's interesting about the centrosome is the centrosome is basically comprised of two components. The first of those components are these yellow colored things. And again, in real life, are they yellow like this? No, of course not. This is just some artistic representation of what this is. But as you can see, there are these two tubular components that are perpendicular to each other, right? So again, perpendicular, of course, means a 90-degree angle. So they're always going to be shown as being 90-degree angles from each other. And these two small tubular components are what are known as the centrosomes. Pardon me, centrioles. There we go. Are the centrioles. What's interesting about these centrioles is one of the ways that we try to find out how things work is that we manipulate it. So one of the things they were able to do is take some animal cells, they were put them under a microscope, and then using a tiny little laser, they destroyed the centrioles. And when they destroyed the centrioles, what they found is the cell could no longer divide. Organizing microtubules is a very important role in the process of mitosis. It helps us to line up our chromosomes and pull the chromosomes to the different sides and all those types of things. And so in animals, they play a vital role in our ability to be able to divide. Awesome. We've unlocked one of the mysteries of life. We were super excited about it. Everybody was high-fiving. And then they were like, just for the heck of it, let's go grab one of those plant cells and put it under the microscope and do the same thing. Right? So just we can really slam dunk it and make it awesome. So they put the plant cell down. They used their tiny laser. They destroyed the centrioles. And guess what happened? The plant cell divided. Exactly. Nothing happened and the plant cell was still able to divide. So why? We don't fully understand. But since every single person in this class, we will talk about spindles and asters more. Aster is just a fancy word for, all right, when we get to mitosis, we will talk about this, but here, I'll do a quick drawing. What's going to happen when the cell divides is that, and I'll just draw this really simply. So here are my two centrioles. They are, again, these little tubular structures that are perpendicular to each other. And what's going to happen is they're going to start to organize the microtubules. What this means is that little lines of microtubules are going to start to form in a star shape around them, kind of like what you're seeing here in the picture. The star-like form that forms around it, aster, right? Aster refers to star. So it's the star-shaped structure. And some of these microtubules are going to become really long, and they're going to connect to our chromosomes, and all sorts of other fun things are going to happen. And again, this is how we organize our chromosomes and move them around from this structure that forms. All right, so these centrioles, as we talked about in animal cells, are vital for cellular division, for this cell to be able to do mitosis. However, the centrioles are only one of the components, right? If you notice also, as we look at this illustration, there is this cloud of material that wraps around the centrioles. This cloud of material is what is known as the centrosome matrix. So this substance or structure, we could use either of those terms to describe it, is a dense collection of cytoplasm. And we give it a fancy name. We call it the centrosome matrix. And again, like I said, it is basically a dense collection of cytoplasm. All right, notice our artist here has done a pretty decent job of representing it. Again, notice this is not a membrane bound. There isn't a membrane around the outer surface holding all of this together. So of course the question comes, what is holding it together? I don't know, hope. I don't know what's holding it together, but we have this dense collection of substances around the centrioles. So our centrosome, the organelle, is comprised of two structures. And those two structures are the centrioles and the centrosome matrix. Now, I know this is when people start to get a little wonky on me, a little confused, but again, this is not a concept you are unfamiliar with. because if you go down into your kitchen, one of the things you will see in there is a table. And that table is made up of two things. It is made up of a big flat board on top and 4 legs. So it is the legs and the board that make the table. It is the centrioles and the centrosome matrix that makes the centrosome. All right, so these are just the two parts that collectively come together to form the centrosome. Now, like I said, this artist did a decent job. This is the one from your textbook, did a nice job of showing us this stuff. But one of the things we need to remember is that every model, every illustration, any of these types of things that we look at are always going to be artistic representations of these things. And so when we look at these artistic representations, we have to remember that these artistic representations are made by artists. And artists aren't always scientists. Now, again, I don't want to be hypercritical, so I won't say that our artists get things wrong, but what we can say is that artists sometimes take a little artistic license with things. And so when you look at representations like this, you always have to take that into consideration, right? Notice the past artists made our centrioles yellow. What color did this artist make our centrioles?
Red.
Right, red or even pink, I would say kind of more of a pinkish, because I think this over here as being more red, whereas this is clearly a little bit lighter, so more pink-like. And what color did they make the centrosome matrix? Did this
Can't really
artist
see.
represent our centrosome matrix? No,
No.
they didn't. All right, so again, a little artistic license. We won't say they got it wrong, we'll just say they gave us a little artistic license, right? This model, on the other hand, again, it's a tiny bit out of focus, but notice right here, we see the two perpendicular centrioles. And now this one represented the dense matrix, but notice they gave it a big, dark, distinct edge, kind of making it look like it is bound in a membrane. Is it really bound by a membrane?
No.
No, it is not. And the other point I want to make, and we'll come back to this picture because it's a little bit prettier. One of the things that I have said many times, and will continue to say many times, is that people often get things wrong, not because they don't know the information, but because they don't read the questions carefully. You've got to read the questions carefully so you can answer them, read them carefully so you can answer them correctly, right? Words matter. One of the important things you need to realize is one of the challenges in this class is there is a lot of information. And with a lot of information, I need to be very precise with my wording to make sure that you give me the piece of information that I want. And I expect you to be equally precise in your answering of it. Let's use the example we just literally finished talking about, because this will be useful for this. Let's say, for instance, on an exam, you saw, and let's go ahead and see if this works. This picture, and I guarantee this picture will be on your exam, and it had a nice big fat arrow pointing right there. And I asked you to identify the organelle. What would your answer to that question be? Excellent.
Centrosome.
Centrosome. Notice exact same picture, exact same arrow, but if instead I ask you to identify the structure, what is your answer there?
Centriole.
Centriole. Excellent. Let's change it up. Identify the organelle.
Nucleus.
Nucleus. Excellent. Identify the structure. This one wasn't as easy, was it?
Nuclear envelope.
Nuclear envelope. Excellent. Identify the macromolecule that forms this structure. You get partial credit for lipid. Ah, there you go. Phospholipid. Excellent. Phospholipid. Kylie got it first. Excellent. How are the macromolecules in this structure arranged?
Bilayers.
Bilayer. Excellent. How many layers of macromolecules do we have here? All right, I'll give you 4 layers, or I'll give you 2 bilayers would work as well. Excellent. Notice my arrow hasn't moved at all. What I've changed is the word that I used in the question to prompt you to the correct question, the correct answer. Now notice also, I could take this and move it just slightly a little bit and now ask you to identify the space. And what's your answer there?
Pore.
Yeah, although I would prefer nuclear pore, but on the first test, I'd probably accept pore. Right? Excellent. My goal is not to be tricky. My goal is to be precise. I will use very precise wording. I'm very careful about how I word my questions, and I want you to be equally precise in your answering of them. I know at first that seems challenging, but the flip side of that is there's a huge advantage to it for you as well. Right? Because let's move the arrow. If I am pointing at something, and it doesn't matter what on here it is, if I ask you to identify the organelle, is nuclear pore ever going to be the right answer for that?
No.
No.
No, right? If I'm asking you to identify a space, is centriole ever going to be the right answer for that?
No.
Is centrosome matrix ever going to be the right answer for that? Is chromatin ever going to be the right answer for that? In fact, I've only given you 1 space so far. So if the test was tomorrow and this is all the material I asked, if I asked you to identify a space, would you even need to look at what the arrow is pointing at?
No.
No, because I've only given you 1 space that you're responsible for so far, a nuclear pore. So it works both ways. I will be equally precise, but that precision not only forces you to be precise, but it also should help you to narrow down the correct answers. And let's finish it off with some of the other things we've learned. Identify the substance.
Nucleoli.
Chromatin.
Chromatin, excellent. Identify the macromolecules that form this substance.
DNA and protein.
DNA and protein, excellent. Identify the structure.
Nucleoli.
Well, although it's just 1, so really it should be nucleolus. Now, honestly, for this first exam, if you wrote nucleoli, you should know the difference between singular and plural. But if you wrote nucleoli, I would give you that credit. Now, actually, that's a good point. One of the things that you'll see on the exams sometimes, because again, when people study from their exams, I think people spend too much time looking at the things they got wrong, which is a good thing to do, but they don't look at the things they got right. So one of the things that I will do on the exam is there are some questions that I will give you an answer of okay to. If I give you the answer okay, then that is my way of telling you that you are getting full credit for this, but it's not really the right answer. I understood where you're going with this. You kind of have the right point. You got it right enough to get full credit. But because this class is cumulative and you need to know this stuff moving forward, know that this answer is not 100% correct. So that's what those 2 letters mean. So if you see that on one of your answers, know that, yes, I got full credit on this, be happy about that, but there's something about this that isn't quite right. So make sure you check and see what that is. I think I posted some of these pictures on Canvas under your modules. The other place, remember, that you can go to see a lot of these things. I don't don't want that up anymore. Give me a second here. So remember here in your modules, yes, you're right. I would put multiple arrows and then the correct answer would be nucleoli. Identify these structures, plural. We're done pretty much with that. Under the study tools, we at ARC are in the process of trying to put this together. We have a few more resources, so it's taking a little bit more time. But Cosumnes River College has this great virtual anatomy lab. And if you go to this great virtual anatomy lab, one of the nice things they have here is they have pictures of a lot of the models and the materials. So look, lo and behold, here is that same model that we were just looking at. And what's cool about this is they will show you a picture like this. And then when you click on the next picture, it shows you the labels. Right, and so here's some light microscopy, some different light microscopies and stuff like that that are in here. So they've got some good tools, some good resources that are on here. And the other thing you can do is you can actually go through the different slides that are all labeled here and see what they are. So you can see some of those things that are here. So things like this cell model are here. That's one of the places you can look at it. Jeff has a lot of these in the open lab. That is something that you can look at for that as well. And as I mentioned, while this picture is going to be on your exam, because it's a good example of a lot of these things, as is this model, which is also the one from the classroom, is that the goal isn't to memorize these pictures. So this isn't going to be the only picture that I will use to show, for instance, the centrosome, or this won't be the only picture I'll use that uses the centrosome matrix in it, for instance. I have no problem using novel pictures. Often when I'm making an exam, if I need something, I go to the almighty Google and I ask the almighty Google, Google, I need a nice picture of a mitochondria. And if I find a nice model picture that shows that, I'll use that. The goal isn't for you to memorize the pictures in your lab manual and your textbook and any other resource like that. Your job is to learn this material. And so as long as it is an obvious example of something, then I have no problem using it on the exam. All right? So you have these resources, and again, there likely will be on the exam. And again, here's the other challenge with this. Remember, normally when I make an exam, it's one exam and you all take the same exam. Remember, every single person in this class is taking a different exam because every person when they log in is going to be randomly assigned questions. So some of you might have 5 questions off of that picture. Is it possible that on your exam that picture doesn't show up? Yeah, unfortunately it is because they're randomly selected. So that, you know, there's no guarantee that you are going to see it. And so how many times you see it is going to be completely random. It's one of the frustrating things about this process in this is that not everybody's getting the same test. What that means is some of your first lab exams are going to be harder than some other people's. Some people's first lab exams will be a little easier. Some's going to be a little harder. The nice thing with 11 exams is that over the period of that, hopefully with randomness, the way randomness works, is that everybody gets about the same amount of challenge in this class. Some people's first exam will be harder and their second exam will be easier. Other people's first exam will be easier and their second exam's harder. In the end, it should all even out, but that's the problem with randomness, right, is that it's random. So it's, again, it's one of the problems with this format, but we're just doing the best that we can with it. All right. Questions on that? All right, our centrosome is our first non-membranous organelle, but it isn't our only. The next one we want to talk about are extensions of the plasma membrane, extensions of the cell. We keep drawing the cell, and I keep drawing it as a circle. Remember, it doesn't have to be a circle. Let's go ahead and cheat and draw it as a square. Because if I cheat and draw this as a square, then one of the things that I can do is I can put extensions on the surface by doing something like this. Right? These extensions of the plasma membrane, and there are primarily three main types. The three main types of extensions are first, what we call cilia. And cilia are finger-like extensions of the plasma membrane. And the second one are microvilli, which are finger-like extensions of the plasma membrane. Well, how can they both have the exact same definition? What the heck's the difference between a cilia and a microvilli?
Cilia are bigger.
They can be bigger often, and that's good. I like that. Let's add that. Typically, let's go ahead and use that word typically, because typically, right, means not necessarily all the time, but typically they are longer than the microvilli. But again, it's not always. And if you don't have them side by side, that's going to be a little bit subjective. Oh, hold on. Looks like I missed a question here. Hold on. If you're asking about the space or organelle, would those be questions on the lab or lab? So typically identification is going to be lab type questions. And again, you guys will see that when you take the practice exam. However, what you have to remember is that this class is all one class. So lab and lecture are just different modalities for asking, for learning information. So on a lab exam, I could point to an organelle like the centrosome and ask you to identify it. But I could also ask you what its function is as well. But yeah, mostly lab exam is going to be identification. So yes, so those are questions like that. Space or organelle or structure are typically the lab exam. Excellent. Right. You guys hit it on the head. Absolutely. And which one is the motile one, cilia or microvilli?
Cilia.
Cilia. Excellent. Cilia are motile. Right? They move. Right? Your book, and this is a great example of one of the things that I talked about before. Your book actually does an amazing job of describing the process by which cilia move. It is this wonderful 7-step process where they describe the whipping motion of the cilia. Really interesting, great knowledge. I'm not holding you responsible for it on the exam. Right? We're not going to talk about it here. It's not on any of your homeworks. If you're interested, knowledge and knowledge's sake, then go ahead and read it. But I guarantee it's not going to be a question on the exam. But what we do need to know is that cilia move. They move in a wave-like motion. Now, do they do this to move the cell through space? Do they cause the cell to move when they do that whipping motion with those cilia?
No, no.
No. Instead, what they do is they move substances along the surface
Yes.
of the cells. Excellent. So they have this whip-like motion, and with this whip-like motion, they are able to propel substances along the surface. It's happening right now as we speak. In your nasal cavity, you have cilia. You are constantly making mucus, and as you constantly make that mucus, the cilia, in this wave-like fashion, move that mucus to the back of the throat, where it either gets swallowed or it gets spit out. Every day, all day long, it is doing this, and we don't pay any attention to it until we have problems with it. Like, say, for instance, you are sick. If you have a cold, one of the things that happens is you overproduce mucus. Right? Anybody who's had kids and ever had kids in daycare know pretty much the first year you keep them in daycare, their nose runs like a faucet because they're exposed to everything. They're overproducing mucus, and those poor little cilia can't keep up with it anymore. Right? Or, and again, I know as hot as it's been recently, it's hard to remember, but back in the winter, right, you go out and sit outside. Back in ancient times, we used to be able to go out and sit and watch football games. And as it's 9 o'clock at night and you're watching that football game and it starts getting cold, those cilia in your nose are sensitive to the temperature change. And as they're sensitive to the temperature change, they slow down. And so as you're out in that cold for a prolonged period of time, the cilia slow down and your nose starts to run. All right, so those cilia are moving substances along the surface. Microvilli, on the other hand, are non-motile. Now, if they're non-motile, then what are their functions? Excellent, I like that. They add surface area. And that surface area is typically going to be important for absorption, and secretion. The cells inside of your small intestine.
What does add surface area? What does it mean?
So I was just going to explain that. So
okay, sir.
In your small intestine is the primary organ that is responsible for absorbing that cheeseburger you had for breakfast. And so if you had a square cell with a flat top, then that cheeseburger you had for breakfast would only have that flat surface of the cell to be able to get absorbed into the body. But if the cell puts up all these finger-like extensions, then we now have a massive surface area by which surface on the top that we can now is going to be in contact with that food and increases the space over which absorption can take place. Right? Your cell, your digestive system is specialized for the absorption of food, and your small intestine in particular has specializations that increase the surface area over 1,000-fold. Right, and these microvilli that stick up are one example of that. All right, so again, our first one is the cilia. Our second one are the microvilli. They are similar to each other in that they're both finger-like extensions that stick out, multiple finger-like extensions that stick out on the surface of a cell. But their primary differences, one is motile for the movement of substances, and the other is non-motile, just to increase the surface area. There is a third extension of the plasma membrane, and that is a flagellum. Flagellum, excellent. What we commonly refer to as a tail. This flagellum basically also is motile. It actually moves in a whipping motion. So basically what happens, where's my pencil? There it is. It's on a kind of a rotary dial. And what happens is that motor rotates around. And as it rotates around, it whips that flagellum around. Now, does that flagellum whip around to move substances through space? No, what does that flagellum do? Come on, I know you know. What does the whipping motion of that flagellum do to that cell?
Propels it forward.
It propels that cell forward in space, absolutely, right? This moves the cell through space. How many cells are capable of propelling themselves through space?
One.
One, and what one cell is that?
Sperm.
There you go, exactly. Spermatozoa are the only cells with flagellum and so they're the only cells that can propel themselves. Through space. Excellent. All right, I've written this all out. Let's look at the pretty pictures. Cilia, short, hair-like, finger-like projections that move. Microvilli, finger-like extensions that are non-motile. Flagella, long tail-like projections, only found in sperm, whipping us through space. We've done this with all the pretty words. Let's look at the pretty pictures. Here, again, we see some examples of those cilia. Remember, as I mentioned, your book does a great job of actually describing the process by which it does it, but I'm not holding you responsible for that on the exam. But their job is to move substances like mucus along their surface. Microvilli just massively increase the surface area, making it better to absorb or secrete materials. And as we talked about here, we see that singular spermatozoon. And again, spermatozoa are the only cells that are capable of propelling themselves with space by having that long singular extension that moves in a whip-like force and propels itself through space. All right, questions on that? Question? All right, excellent. Let's move to our last non-membranous organelle, and that is our cytoskeleton. As the name would indicate, our cytoskeleton is the skeleton of a cell. There you go, simple and straightforward. What does our skeleton do for us?
Provide a framework.
Excellent. So it provides a framework. Anything else?
Help moving.
Right. And allows movement. So guess what the cytoskeleton does for the cell?
Provides us a framework and allows it to move.
There you go. See how convenient that is? Absolutely. Excellent. Now, all of our cytoskeletal structures do that. All cytoskeletal structures provide framework and allow some type of movement to occur. But one of the important things to know is that there are three main types of cytoskeletal structures. Let's do that. The first one, and again, just like before, we'll do it with the pretty words first, and then we'll look at it with the pretty pictures. The first are what are known as microtubules. I'm sorry, microfilaments. Or what are also known as actin filaments. Now, with a name like microfilament, or more specifically, actin filament, guess what protein is, gravy. Guess what protein is responsible for forming our actin filaments?
Actin.
How about the actin protein? Excellent. So the protein that forms these are actin. Excellent. Actin is a dimer. Anybody know what that is? What's a dimer? No, oh, that's true. It is similar to that, but not exactly in this case. In this case, what a dimer is basically a protein that is made up of two components. So basically, what happens with actin basically is comprised of two globular proteins. That right there, what you see, just two ball-shaped proteins put together, is an actin protein. So what happens is, what we can do is we can add a second actin onto here. And again, my drawing skills are not the best. Well, I'm drawing circles, but you get the idea. And then I can add a third. And then I could add a fourth. And then I could add a fifth. I wonder if it'll be easier if I go that way. Yeah, it's easier if I go that way. And a sixth. And notice suddenly what I've made is a long strand. And what's nice about these actin strands is, even though it wasn't easy for me, it really is that easy to add proteins onto it and off of it. So again, they're going to help form the framework of the cell. But one of the important things about actin is that they are going to be able to allow for dynamic movements of the cell. when we get to, yeah, absolutely. When we get to cardiac muscle and skeletal muscle, actually all muscle, when we get to muscle, we'll see that actin is one of the important contractile proteins that is going to play a really important role in our movement and our contraction of a muscle. But the other place you see actin playing a really important role is actually in the growth of our nerve cells. Remember I mentioned how you have some nerve cells that start in the small of your back and go all the way down to your big toe, right? It's like a meter long. And the way those cells grow is they start in the small of your back, and then what they do is they put out these extensions of their plasma membrane that are part of a growth cone. And these growth cones are formed by these actin filaments that expand out. And what happens is it's testing the environment. If over here in the environment, it comes across something that it is repulsed by, then we can rapidly break down those actin filaments so it doesn't grow in that direction. But if over here we find some attractive cues, then we can rapidly expand our actin and grow in that way. So yes, as mature adults, it allows us to move our body through space, but it's also going to play a really important role in helping the growth of our nervous system, for instance. So it allows for dynamic movements, dynamic changes in the shape of the cell. And let's add that as well. Movement or changes in the shape of the cell. Excellent. All right, I need to get rid of this. So let's cheat and do it that way. Perfect. Excellent. Our second cytoskeletal structure are what are known as intermediate filaments. Now, with intermediate filaments, there isn't just one protein that forms them. So in this case, for proteins, we will put that it varies, because there are multiple proteins that can be involved in this process. Intermediate filaments are what I like to think of as the I-beams. These really play a vital role in the shape and the support for a cell. Think of it this way. Back in ancient times, well, actually, I guess you can still get in your car and drive around. And you can go get in your car and drive around. It's one of the few things you can do as long as you keep your windows open, right? But one of the places you could go is to downtown Sacramento. And as you drive by downtown Sacramento, what you see are these massive, huge skyscrapers with just these big, huge sheets of glass up the entire surfaces of all of the outer surface. And are those sheets of glass what are holding that building together? No, of course not, right? They're what's on the outside, they're what we see. But on the inside, you know there's got to be these massive structural supports, these massive I-beams, right? Those I-beams that are going to play an important role in holding that building together and letting it grow up to the sky and have that beautiful outer surface because it has that massive structural support inside. And that's what those intermediate filaments do. They form that structure and shape. They're that internal support that helps to give the shape and the structure and therefore affect the function of that cell. Lastly, oh, and I should say also the microtubules, the microfilaments are our smallest of our cytoskeletal structures. The largest of our cytoskeletal structures are our friends, the microtubules. Microtubules, as we know, are going to be organized by our centrosome. They are made of proteins called tubulin. These coiled shaped tubular proteins that come together to form these massive structures. Again, they're cytoskeleton. They allow for the framework. They allow for movement. Movement in the cell, let's say it that way. And notice the key that I said there, they allow for internal movement. Again, I have to work on my analogies because normally at this point, what I say in the class is that, right, most of you got here today because you had a car that has a motor inside of it. We talked about motors. What does a motor do again?
Propel forward.
Right, it produces a mechanical force. And what does it use to produce that mechanical force?
Oil. Fuel.
Say again?
Fuel.
Yeah,
Fuel.
fuel, right. A motor converts chemical energy into mechanical energy, all right? And so you had that motor that got you here, but that motor drove along the surface of the streets, like Interstate 80. Now, did Interstate 80 move to allow you to get to campus? No,
No.
of course not, but it provided a surface that your motor could move along. And that's what microtubules do. They allow for internal movement. They basically are the streets, let's put that in quotes, that our motors can move along to move a substance like an organelle through the body. Let's say it this way, around inside the cell. So again, notice the street doesn't change shape. The street doesn't actually move, but it provides the passageway for that motor to move along. And that's what our microtubules do. They move our chromosomes, they move our organelles, they move vesicles, they move all sorts of substances around inside the cell by having little motors walk along their surface. I've done this with the words. Let's do this with the pretty pictures. Here are our microfilaments, or remember what I said, actin filaments here. If you didn't like my drawing, you can see this dimer globular actin subunit forming that strand. Again, these allow for dynamic movements of the cell, change the shape of the cell. Here we see those intermediate filaments. Again, those are the I-beams forming the structure and support. It even looks like a supportive cable, right, holding up a suspension bridge or something along those lines. And here we see those globular tubulin forming these big coiled microtubules that are the highways and byways inside. This is a pretty picture from your textbook. I like this picture too, because it kind of shows them all together. Here we see some of those microfilaments helping to hold up the microvilli, giving them their shape. We have our intermediate structures helping to form structure and support. And then notice even our microtubule here. Here in our microtubule, we have a vesicle, we have a piece of an organelle that are being transported along the surfaces of those, along those highways and byways. So all these things work together to provide the structure and the support, forming the skeleton of our cell. All right, questions on that? All right, those are our non-membranous organelles. Let's spend some time talking about our membranous organelles. Now, actually, we've already talked about one. Let's not forget that the nucleus is a membranous organelle. Right? In fact, remember, it actually has a double membrane. So really, we're now talking about our second membranous organelle. And notice, conveniently enough, I mentioned our nucleus because lo and behold, here is our nuclear envelope. And notice right next to our nuclear envelope, and sometimes even connected to it, is our second membrane-bound organelle, the endoplasmic reticulum. I thought we had talked about this on the first day, but I know I've talked about a lot of things on the first day, so I want to make sure that we are clear on this. Again, on the exams, there is not a word bank that you are getting for the lab exams. You are responsible for knowing these terms, and spelling matters. So on the exam, if I asked you to identify this organelle, you need to say endoplasmic reticulum. If, however, you were writing an essay question about it, once you've written it out once, how could you then abbreviate it then? How do most people? Yeah, ER. Absolutely. So again, ER, you could do it that way. But if on the exam, I showed you this picture and I pointed to that blue thing and I asked you what it was and you just said ER, I don't accept abbreviations. So that I would consider that a spelling error and you would get partial credit. All right. Remember, you get one letter. So you can have an O instead of a U or something like that, or you could transpose an I and a U or something like that, or U and an I, something along those lines. You can have it that way. If you have one typo like that, then I will allow it. But if you spell something more wrong than that, you do get partial credit. And again, 50%. So I don't slice it any thinner than that. When you get partial credit, 50% is what we do. Okay. So let's spell it the right way. So we have that endoplasmic reticulum. Oops, still didn't get it right. There you go. See, I get one letter just like you. All right, excellent. That endoplasmic reticulum. However, endoplasmic reticulum comes in two flavors. If you notice, the part that is in closest proximity to the nuclear envelope has all these globular red dots all over it, right? Haley's already kind of hinted at it. And these red dots make it very rough in its appearance. And so that particular portion of the endoplasmic reticulum, how do we identify it? Well, as Haley already pointed out, it is our rough endoplasmic reticulum, which you would then abbreviate RER. So this down here is our rough endoplasmic reticulum. And these globular components that are on its surface are, oops, hold on, we'll get to that in a second. Whoa. Are our ribosomes. Now notice, this organelle is bound by membrane, and it's these flattened interconnected sacs of membrane. We have a fancy name for that because, again, we have a fancy name for everything. And that fancy name is cisternae, being the plural. And of course, the singular is cisterna. So these flattened interconnected membranous sacs are the cisternae. And for the rough endoplasmic reticulum, those cisternae are covered with ribosomes. Now, go ahead.
So the whole structure, regardless of having the smooth ER and rough ER, so this is the ER, right? Endoplasmic reticulum, right?
Correct. However, on the exam, you are going to want to specify the difference between the rough endoplasmic reticulum and, as you've already pointed out, this other one that doesn't have the ribosomes, which is the smooth endoplasmic reticulum. So honestly, I can't think of a question where the answer to it would just be endoplasmic reticulum. You are going to want to specify the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum. Now, both the smooth and the rough are made up of flattened interconnected membranous sacs, so both are made up of cisternae. But the difference is the rough endoplasmic reticulum have ribosomes on them, and the smooth endoplasmic reticulum does not have ribosomes on. All right?
Yes,
So
sir.
the big difference between the rough and the smooth are the ribosomes. So then I guess the big question is, what is it that ribosomes do? What is the function of these ribosomes? Excellent. They make proteins. Excellent. Right. Remember, as I mentioned, when I teach this class in 102, I like to use the analogy that a cell is a factory. And the job of that factory, for instance, is to make shoes, right? So Nike's got a factory and its job is to make shoes and distribute them to the world. Well, if that's the case, that makes the ribosome the 8-year-old Chinese kids. right? Because they're the ones who are putting the shoes together. They're the ones making the proteins. And that's their job. Those ribosomes make the proteins. And if you think about it, that makes sense why it would be right next to the nuclear envelope, right next to the nucleus. Because here in the nucleus is where we have the instructions to make the proteins. And so those instructions come out the nuclear pore and come right to the rough endoplasmic reticulum, right to the ribosomes, where they can then be made into proteins.
Professor,
Yes,
did you say the smooth have the ribosome or it doesn't?
does not. Rough has the ribosomes. That's why it looks rough. It has a rough appearance because it has the ribosomes on it. The smooth endoplasmic reticulum is smooth because it doesn't have ribosomes on their surface. Now, one more thing. Not all ribosomes are bound to the rough endoplasmic reticulum. There can be some free-floating ribosomes just in the cytoplasm. So there are going to be some ribosomes that are just free-floating in the cytosol, in the cytoplasm of the cell, but most are bound to the rough endoplasmic reticulum. But you did make a good point. If the smooth endoplasmic reticulum doesn't have ribosomes, then guess what it absolutely positively cannot do? If it doesn't have ribosomes, it can't make proteins, absolutely. So what does the smooth endoplasmic reticulum do?
Synthesize
Synthesize what?
lipids and steroids.
Well, lipids are a type of steroid.
Yeah.
So yeah, it can be used to make lipids. It also can break down lipids. Excellent. So it can make and break down lipids and other fatty acids, things along those lines. And also in some cells, and again, muscle cell will be a great example of where we'll see this, it can actually be specialized for storage, where it will actually, like you said, in our muscle cell, to contract, we need calcium. Calcium makes cells do wonky things. And the smooth endoplasmic reticulum in our muscle cells actually stores a massive amount of calcium. All right? And again, hopefully this is an obvious question. Where is most of the calcium stored in your body?
Bones.
Yeah, in the bones, absolutely, right? 99% of your calcium is stored in the bones. However, outside of the bones, the smooth endoplasmic reticulum of our muscle cells, our skeletal muscle cells, is the second highest concentration of calcium in the body. So we store massive amounts of calcium in there because contracting is a very wonky thing. And what does calcium make cells do?
Strong and dense. Make it dense and strong.
Well, that's what it does to bone, right? Absolutely. But in general, to cells, calcium makes cells do wonky things. Absolutely. Weird's not bad. I prefer wonky. All right. Questions on those two? All right. So go ahead.
Can you go back to the cisternae? Is that
Yes.
just the turns and that formation of it?
Yes. So exactly. Absolutely. Again, it's about labeling things, right? A flattened membranous sac that is forming these organelles is what we call a cisternae. So cisternae is the name for a flattened membranous sac. Here we have it two ways. We have it with ribosomes on it and without ribosomes on it. But guess what? This isn't the only cisternae we're going to see. We see cisternae in another organelle as well. And that other organelle that we see it in as well is the Golgi apparatus, or what is also known as the Golgi complex. Notice once again, we have flattened membranous sacs. And guess what we call these flattened membranous sacs?
Cisternae.
Cisternae, absolutely. Cisternae, excellent. Oops, wrong place. Excellent. Cisternae are flattened membranous sacs. The big difference here in the Golgi complex is that the cisternae are not connected. Remember, in our rough and smooth endoplasmic reticulum, the cisternae are connected. Here they are not. Basically what it is like a stack of pancakes. In fact, that's often how you will see it illustrated. When you see it illustrated, basically you see these flattened membranous sacs that are not connected to each other, kind of like a stack of pancakes. The reason for this is the function of our Golgi complex. Our Golgi apparatus's job is to modify our proteins, make them functional. And it does this in a couple of ways. One of the ways it does it is by cutting and folding the proteins. And the other way that it does it is by, remember we talked about the quaternary structure of proteins. We take subunits and put them together.
Functional.
Yep. So we're going to put those subunits together to make a functional protein. But that's only one of the functions, right? One function is to modify our proteins and make them functional by adding those subunits together. But the second thing our Golgi apparatus is going to do is package the proteins to ship them to their final destination. I'm going to cheat and take one of my cisternae here and move it down here to the bottom so that I have a little bit more room to play through. The way this happens is that proteins move through the Golgi apparatus, and I'm just going to abbreviate it here for space, in one direction. So I need to make this a little bit smaller and then move that there like that. Excellent. Proteins move through the Golgi apparatus or Golgi complex. Again, both of those terms are acceptable terms in just one direction. So they enter one side and they exit the other. Right? The side that it enters from is what is called the receiving side, or what is also known as the cis face. And then it exits out what we call the shipping or mature side, or what is known as the trans face. So proteins go through this in just one direction. And as they work their way through this, they are modified, they're bundled together, they're packaged, and ultimately sent to their destination. Now we'll talk a little bit of how that process goes, but we also need to talk about is how they move from one cisternae to the next, and from that cisternae to the next one, and so on and so forth. And as you can see from this illustration, the way they do that is with vesicles. So they're one of the ways you're going to be able to easily identify the Golgi apparatus is there's always going to be a ton of vesicles associated with them. Now, let me show you actually one more thing I want to mention here, and then I'll show you the pretty pictures that go along with this. Another way that can help you to identify the Golgi complex is because proteins move in just one direction, that receiving side, the cis face, always faces the nucleus, and therefore also the rough endoplasmic reticulum. And then the trans maturing face always faces the plasma membrane. Oops. Faces the plasma membrane. All right, so I've made a mess of the board. Let's take a look at the pretty pictures for this. So again, its job is to modify the proteins and the lipids as well, modify those, package them up, and send them to their ultimate destination. And part of that process, they can actually modify our plasma membrane. Let's look here to see how transportation occurs. So you've got the right idea. The Golgi apparatus actually takes the sequence of amino acids, that first level, again, level, not layer, protein, where it's a sequence of amino acids. And then what it does is it cuts it, it folds it up, and then it puts all the subunits together. So yes, all our ribosome does is makes the string of amino acids. It just makes the single strand of amino acids, which, remember, is our primary level of protein structure. And then you're right, it is the Golgi apparatus who cuts it, and that allows the secondary structures to form. That gives it its three-dimensional structure, and often it is adding multiple subunits together to make the fully functional protein. And remember, not all fully functional proteins are globular, but you've got the right idea. It is going to make the fully functional protein as a result. This processing occurs within the different layers of the cisternae. And so notice here with this picture, what we see really nicely is here we have some proteins that are inside of the cytoplasm, inside of the cisternae. And here we see some that are bound to the plasma membrane of the cisternae. A small piece of the plasma membrane buds off. And when it buds off, it pinches off. And notice it can have proteins on the plasma membrane, or it can actually have proteins inside. Once it completely pinches off, then as a result of that, it becomes a completely free-floating vesicle. A vesicle is just a fancy name for a small piece of plasma membrane that has proteins either inside it, on it, or both. Now, in this case, our free-floating vesicle goes to the next cisternae, where it attaches to it. Notice when it fuses to it, it becomes part of its plasma membrane. Any proteins that were on its surface are now on the surface of this new cisternae, and any proteins that were inside of it are now released inside of the cisternae. And so this occurs through each layer, and through each layer, these proteins are modified. Now, the other place we see this really nicely is this picture here. Notice this picture here, we can see that one side is facing the nucleus and the rough endoplasmic reticulum. One is facing the plasma membrane. And again, those two faces are the forming face, or the cis face, and the one where you leave, the exit, the maturing, or that trans face. All right. Now, remember, and where can I sneak this in? Actually, let's do this way. All right. Everybody seen this pretty picture? Let's redraw this in a less pretty form. Here is my plasma membrane, and let's actually make this black. There's our plasma membrane. Here are two cisternae of our Golgi apparatus. And our goal now, actually, you know what? I'm going to cheat. Let's move these here a little bit closer, and then I can write over here. Excellent. Remember, our Golgi apparatus has two functions, two main functions. It modifies and forms the protein. But remember, it is also going to ship the proteins to their final destination. And essentially, there are 3 final destinations for a protein. Let's see an example of this. Let's take an example where we have a vesicle that comes off of our Golgi apparatus. It leaves, let's change to red now. That vesicle leaves the final trans face of our Golgi apparatus. And in this particular case, this one has proteins inside the vesicle. And I need to make this a little smaller so everything fits. So this one has proteins inside the vesicle. So let's make some proteins. Protein, 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 protein on the inside. All right. Now, what's going to happen to this one that has proteins inside the vesicle is it is going to carry this vesicle to the plasma membrane. So it travels to the plasma membrane. And it fuses to the plasma membrane. So let's sneak that up here. When it fuses with the plasma membrane, and this time I will use red for its, it fuses to the plasma membrane, becomes a part of the plasma membrane. And when it does that, what happens to all of the proteins that were inside of it?
Do they escape?
Yeah.
Spill out.
They are released from the cell. So when this binds to the plasma membrane, those internal proteins are released from the cell into the extracellular fluid. Fluid. We have a fancy name for this process where substances are brought to the plasma membrane of a cell and released from that cell. Anybody know the fancy name for this process? There you go, exocytosis. So also known as exocytosis. There you go. So the first possible destination from a for a protein is the protein, and let's make this big again, can be released from the cell via exocytosis. Excellent. Questions on that process? And I'm going to cheat. Do that. And then that. Perfect. So that we can tell them apart. All right. Again, in this case, the vesicles are just moving throughout. So again, remember we talked about vesicles can often be moved along the surface of microtubules. Motors can be moving them along on the inside, and that's one of the ways we can bring them to the surface, to the plasma membrane to be released. They move along the surface of them, yeah, absolutely. Yes, although honestly, it's more like a sidewalk. What's really cool, they've actually looked at these motors under the microscope, and when they look at this under an electron microscope, what they actually see is these motors actually kind of have these feet-like structures, And what happens is they kind of grab onto the, or hands, I guess kind of like climbing a ladder would be another example, like climbing a ladder. They're able to grab onto the microtubule, and then as they do that, they swing forward. And so they really do walk along the surface of that microtubule, or climb the microtubule like climbing a ladder, absolutely. Yep. Of course, I need to get rid of that now, but you are absolutely correct. That is an accurate description. All right. Any other questions on exocytosis, process 1? All right, so let's talk about process 2 then. Again, we have a vesicle that leaves the transphase, maturing phase. Only this time what happens is the proteins are on the surface of the vesicle. Or let's be more specific, on the plasma membrane of the vesicle. And that, of course, is way too big, so let's make that smaller. Excellent. So in this case, our proteins are on the surface of the plasma membrane. Once again, this vesicle is going to migrate to the surface of the plasma membrane. And once again, when it gets there, it is going to fuse with the plasma membrane, become a part of the plasma membrane. However, this time, the proteins are on the surface of the plasma membrane. So when this fuses and becomes a part of the plasma membrane, notice what's going to happen is that these proteins are actually now going to be on the surface of our plasma membrane. So as a result of this, in this case, these external proteins are not released from the cell. Instead, they are placed on the surface of the cell, right? On its plasma membrane. Of course, we have a fancy word for this as well. In this case, we see these proteins are expressed on the plasma membrane. Now, this may seem somewhat silly or not important, but it depends a lot on what these proteins are. Some of these proteins could be channels that allow ions to come into the cell that weren't able to come into cell before. Or maybe it's a receptor so that a hormone can bind to it, and that hormone can now have an effect on this cell that it couldn't have before. By putting a protein on the surface of the cell, we can potentially cause a permanent change to this cell. Cause it to do things that it was never doing before. So being able to express proteins on the surface is actually a really, really powerful process. And so, of course, that is our second possibility, our second possible destination for a protein, is that the protein can be expressed on the plasma membrane of the cell. And I want that in black, and I want that in bigger. All right. Question on that. All right, excellent. So let's talk about the third possible destination for a vesicle. So again, we have a vesicle that leaves. And what's cool about this particular vesicle is it can have proteins on the inside. It can have proteins on the outside. It can have one, the other, or it can have both. So in this case, and actually let's write it here. Here, proteins can be inside or let's say it this way, and or on the plasma membrane. So now I want that green and I want it small. Now, notice for the first two vesicles, they went to the plasma membrane. But guess what? Our vesicle doesn't actually have to go to the plasma membrane. In this case, what happens is instead of going to the plasma membrane, the vesicle stays inside the cell. And when it stays inside the cell, it serves some type of function. Notice in this case, we have a membrane-bound structure that has proteins on the inside or the outside that is serving a specific function of the cell. Essentially, it becomes an organelle inside of the cell. And so that's our third possible destination for a vesicle. Our third possible destination for a vesicle is that the protein can become an organelle. Oops, not sure why that's all in caps. And there you go. Those are the three possible destinations for a protein, really for the vesicle, as it leaves our Golgi apparatus. Now notice, I've done this with my incredibly horrible, really simplistic drawing, but if we go back to the pretty picture from your textbook, notice we see all three of these processes. Notice here, oops, wrong button. We see exocytosis, where that vesicle has the proteins inside of it and it releases. Notice we see this pathway here, where the proteins are on the plasma membrane of the cell, and we are forming a part of the plasma membrane, allowing us to change the plasma membrane of the cell, or it can become a free-floating vesicle that is going to have some job of its own. So this picture from your textbook does a nice job of showing those three pathways that we talked about for what happens with our proteins once they are packaged up. All right. Questions on that? All right, excellent.
Can you hear me, sir?
I can hear you now, yes.
Yeah. On this structure or picture, we see some terms like phagosome. Phagosome?
What?
Phagosome?
Yes.
So what's the role? What role is played by phagosome in here?
So that leads us to this lysis, to this vesicle, this organelle. We're talking about organelles, and that's what we're going to talk about right now. The short answer to your question, a phagosome, what a phagosome does, and let's go back to our previous drawing. This might be the easiest way to describe this. So we talked about the process of exocytosis, right? We're comfortable with that idea?
Yes.
So what some cells are capable of doing is that, say, and what color haven't I used yet? Let's use purple. I like purple. Let us say that there is a virus that happens to be hanging out here outside of the cell. One of the things this cell is able to do is attach that virus to its plasma membrane. When it attaches that virus to the plasma membrane, it is actually able to wrap its plasma membrane around that virus and actually bring it inside of the cell. So what would end up happening is we would have a piece of plasma membrane that would have that virus inside of it. That make sense?
Yes.
What would we call that process?
Phagocytosis.
True. Specifically, this process is indeed phagocytosis, where it is bringing it in, or part of phagocytosis. But the simplest form, when we grab something and bring it in, it would be endocytosis.
Mhm.
Right? But you're absolutely correct. Specifically, if it's something harmful, that harmful thing is, we would call that process phagocytosis. Right? So you're absolutely correct. So specifically, this would be phagocytosis to bring it in. And this vesicle with the virus inside of it is what we would call a phagosome. So one of our free-floating vesicles here is actually able to bind to that phagosome. And when it binds to the phagosome, it releases digestive enzymes. And those digestive enzymes break up and destroy that pathogen. And any idea what we would call that particular type of vesicle? We would call it a lysosome.
Lysis?
Lysosome is what we would call it. We would call it a lysosome. So notice, if we go back to lecture, there are dozens of different types of vesicles that can be found inside of the cell, vesicle type organelles. I'm going to hold you responsible for the two most common. The most common one are our lysosomes. Lysosomes contain hydrolytic enzymes. And what does a hydrolytic enzyme do again?
Break water.
Well, it uses water, it doesn't break water, but it uses water to break bonds.
Yeah.
Absolutely. So one of the things these things can do, like I said, is they could bind to that phagosome and break down that virus. Does that mean lysosomes have no function if you're healthy? No, it turns out they can be useful when you're healthy too, because as we said, cells make a lot of proteins. And proteins can be very important and useful for something, and then they're not so useful anymore. They're kind of like Christmas decorations. Every year you got to put out your Christmas decorations, but come January 1st, you got to put them all away. And lucky you have that attic or that space in your garage or those boxes in your closet where you can put those things. But they take up a lot of space and they're a pain in the butt. Right? And cells, do cells have a lot of space to store things that they only use once a year?
No.
No, right? Or like my daughters, right? My daughters love playing with Legos. We have a room dedicated to Legos. So they take the Legos, they put it together into building this big, beautiful house. And when they're done making that house, they come to me and say, now I want to make a boat. Do I run out to buy them new Legos so that they can make a boat?
No.
No, of course not. I take the house, I smash it on the ground, and now they can reuse all of those Legos to make a boat, right? Cells are the ultimate recyclers. They do the same thing. We don't want to store any of these extra proteins inside of them or organelles that have been broken down or are damaged, right? Proteins that are no longer useful. So what we're able to do is we're able to break down those unused or damaged or dead components and recycle them. Free radicals, no. We'll talk about free radicals in a later, and DNA, no also, because remember, DNA is inside the nucleus. These lysosomes are in the cytoplasm. So lysosomes can't get to the nucleus. Lysosomes don't break down free radicals, but luckily, we do have a membrane-bound vesicle organelle that can break down those free radicals and detoxify a cell. And those are our peroxisomes. So peroxisomes are the ones that break down fatty acids. Again, as well as the smooth endoplasmic reticulum, they break down organic compounds like free radicals, and they detoxify the cell. So these are two examples of vesicle-bound organelles. Yes. Although, again, you got to be careful with the peroxides as well, right? Two chemists walked into a bar. One said, I'll have H2O. The other said, I'll have H2O2. Second one died. Right? You see, even chemistry jokes are horrible as well, right? God, chemistry is such a horrible thing. Nothing good has ever come from chemistry. It's not even a real science. All right, excellent. Questions? Questions on that? All right, excellent. So we do have one issue though, right? Let's cheat for a second and go back. All right. Notice we were eyeballing this before, and we were talking about and identifying things that we needed to know. So we've already identified some of these things. Just out of curiosity, identify that structure.
Golgi complex?
There you go. Cisterna. This here is a cisterna. However, if I instead asked you to identify the organelle, what organelle is this?
Golgi apparatus.
Golgi apparatus, absolutely, right? We can see the little bit of vesicles that are associated with it as well. Conversely, oops, no, hold on, didn't want to do that. Identify this structure. Or maybe this one's easier. Identify this structure. Also a cisterna. Excellent. Notice structure, structure. The answer to those are the same. However, organelle, what was this? Identify the organelle. Golgi apparatus. What organelle is this?
Endoplasmic reticulum.
And you get partial credit for that on the exam. What'd you forget? Smooth. Excellent. What is this structure?
Rough endoplasmic reticulum.
No.
Oh, that's the
What is
organelle.
Structure, exactly. Cisterna. What organelle is this?
Rough endoplasmic reticulum.
There you go. Identify the globular structure here in red.
Ribosomes.
Ribosomes. Notice this model does a pretty good job of showing us, and we're able to figure these things out. We could figure out that this was the rough endoplasmic reticulum, this is the smooth endoplasmic reticulum. Now, as we've talked about, should the smooth be closer to the nucleus than the rough? No, because remember the rough endoplasmic reticulum has got to be able to receive the information from the nucleus so that it can make it. So they've got their rough endoplasmic reticulum a little bit further out, but we can clearly see the difference from them. And again, not an error, just a little bit of artistic license. Notice their other big artistic license. Any idea what these red structures here are? Identify those red globular structures. More ribosomes, absolutely. Free ribosomes. Remember, we said free ribosomes float around inside the cytoplasm. Now, do they congregate together like schools of fish throughout the cell? No, of course not. So, but again, a little bit of artistic license that is taking place here, right? So notice, because of the structures, you know, we can tell the nucleus, we can tell the centrosomes, we can tell the difference between the Golgi apparatus and the rough endoplasmic reticulum and the smooth endoplasmic reticulum. All of that we can kind of be seen. But notice we also have some vesicle organelles, purple ones and yellow ones. Now, luckily, this is a representation of the cell. So all we need to know is in a real cell, which one is purple and which one is yellow. Is a peroxisome purple or is a lysosome purple in a real cell?
Neither.
Exactly, neither of them, absolutely, right? Neither of them are purple and yellow. Those aren't the two colors of them. That's just the two colors that our model maker here has used to represent. The problem is they're both just vesicle organelles. So notice with the rough endoplasmic reticulum, it has the ribosomes, it has the cisternae, it has characteristics that we can use to identify them. Is there really a characteristic about these that really jumps out as being different between these two? Well, Haley, that is the question that I'm getting to, absolutely. There isn't, notice, something that really helps us to tell them apart. Size, you are absolutely, is actually right. That is the one key difference. It turns out in real life, lysosomes tend to be larger than peroxisomes. So in real life, that happens to be the case. And you are correct. That is kind of the only distinction in them here. What we are going to do is we are going to make up a rule by convention. Remember, a rule by convention is just a rule that a bunch of people get around and decide. This is the rule, and we're going to follow this rule, right, under whatever society, right? That by convention, Pluto was no longer a planet, and then it was a planet, and then now it's just a planetoid, and back and forth and all of that kind of stuff, right? These are rules that a bunch of people get together and make up. Well, we're going to make up a rule here as well. A rule we're making up here, you're right. It would make sense. It would be easier to cheat and go P peroxisomes or P purple. But as someone pointed out, on this model, the purple vesicles are larger. And since lysosomes are larger, the rule we're going to set up in this class is that the purple vesicles are the lysosomes. And the yellow vesicles are going to be our peroxisomes. So that is the rule we are going to make up in this class. Oops, I misspelled vesicle. I don't know. Oh well. You get the idea. That is the rule we are going to make up because on this model, that will be the case. Now notice, conveniently enough, this model also has purple vesicles and yellow vesicles. This is the second model that's in our classroom. Here we can again see those centrosomes that we talked about, the centrioles in our centrosome. We see yellow vesicles. Now notice sometimes the yellow vesicles are smaller, the purple are larger, but since we set up a rule, we're going to stick to that rule. The purple vesicles are going to be lysosomes, and the yellow ones are going to be our peroxisomes. Now let's take a good look at this model. This model, remember, was the one that did a nice job of showing us our centrosome matrix. But any idea what organelle this might be?
Microvilli.
Well, but would microvilli be on the inside?
Yeah.
What if I did this? If I cheat and do that, does that help a little bit? You see these, you see vesicles. What is this supposed to represent? Yeah, it's supposed to represent the Golgi apparatus. Now, should the Golgi apparatus, A, be connected to each other, and B, be connected to the nucleus?
No.
No, they've done a horrible job with that. And look at what else they've done a horrible job. Like, no, they did a great job of putting our rough endoplasmic reticulum right around our nucleus. But then they made that rough endoplasmic reticulum like a water slide that goes all the way to the outside of the cell. And here's another one that goes all the way to the outside of the cell, right? And here's just a random piece connected to the outside of the cell. And notice, as far as the eye can see, we see white spots representing the ribosomes everywhere. In fact, if you look closely, and I have, you will see that the only cisternae that doesn't have ribosomes on it is this one right here. This little one here connected to the outside is the only one that doesn't have ribosomes on the outer surface. So, if I'm going to, on the test, ask you for the smooth endoplasmic reticulum, is this the model that I'm going to use?
No.
No, absolutely not. Remember, as I said, I will always use obvious examples of things. This is not an obvious example. It's a horrible example of the smooth endoplasmic reticulum. And as I've said many times, what I like about this class is hard. I don't have to be tricky on this because it's going to be hard anyway. So I don't have to be tricky. I can be very straightforward. So I always use obvious examples. So I clearly will never use this for the smooth endoplasmic reticulum. That doesn't mean there aren't good things on here. We saw the centrosome matrix. It's got a nice nuclear envelope. Chromatin is very clear on this. It's got this great, huge red nucleoli in here, right? Whatever these orange things are we haven't talked about yet are in here. Those are really good on here. There is some good stuff on here, right? And remember, I use this example because this is one of the two models. These two models are the models that are in the class. And like I said, on the exam, we definitely use the stuff that is in the classroom for this. But remember, I just don't want you to memorize these pictures. So one of the things that I'll use is they did actually update this model. They realized, oh, this model is really bad. We should fix it. And here's the fixed version. Notice they did an excellent job of fixing our Golgi apparatus, made that much more obvious. But notice we still have our slippery slide of rough endoplasmic reticulum. This is still the only smooth endoplasmic reticulum. So again, they messed this one up. But the point that I usually make is that I would have no problem showing you this model and this picture right here for the Golgi apparatus, even though this isn't a model that's in the classroom. Because remember, your goal is not to memorize pictures. Your goal is to understand this information. All right? So again, I'll always use obvious examples, even if it's a novel one you've never seen before. As long as it's an obvious example, I have no problem using it as something that you're responsible for, because your job isn't to memorize a bunch of pictures. Your job is to understand this information. And read the questions carefully so you can answer them correctly. If I had an arrow pointing here and I asked you to identify this structure, and you said that this was the rough endoplasmic reticulum that has ribosomes on the outer surface, and its primary job is to take information from the DNA inside the nucleus and use it to make a sequence of amino acids that is going to be sent to the Golgi apparatus. Well, all of that information is 100% correct. Did you answer my question? No, my question was to identify the structure. And what structure is that? Cisternae, excellent. All right. So again, read the questions carefully so you can answer them correctly. People often lose points, not because they don't know the information, but because they don't read the questions carefully. That is always important, but it is especially important here. Because in the classroom, when we're taking a lab exam, if you're not 100% certain on what a question is asking, you can raise your hand and I can come around and I can word it a different way to make sure you understand that. Can you do that on this test? No. So again, I've worked very, very hard to be very precise, very specific, very standard in the way I ask my questions, because I don't want my asking of the questions to get in the way of you showing me you have the information. All right? So I am very particular, very precise. I try to be very standard in the way I ask my questions so you know exactly what piece of information I'm looking for and you can give it to me. All right? The problem is you don't read the question carefully. People get all excited because they see a picture like this and they see the arrow pointing to something like this, and they get all excited. I know what that is. That's a Golgi apparatus. And they write down Golgi apparatus without reading the question. And maybe the question was, identify the structure. Or maybe the question was, give me the function of this organelle. Make sure you read the questions carefully so you can answer them correctly. All right? Questions on that? All right, I thought this was a good place to do this, but we're actually not done with our organelles. We need to cheat and go back a little bit. We have one more membrane-bound organelle we need to talk about, and that is our mighty mitochondria. The mitochondria are special in a couple ways. One of the ways they're special is they also have a double membrane. What was the other organelle that had a double membrane again? That's all right. I wasn't paying attention either.
Nucleus.
Nucleus, excellent. The nucleus was as well. Notice this double membrane is a little bit different. It has a smooth outer membrane, and then it has this elaborately folded inner membrane. And this elaborately folded inner membrane has a special name. That special name is a crista, the plural being the cristae. And again, cristae is similar to cisternae, but it's not the same thing. Cisternae is a flattened membranous sac, whereas a cristae is a folded inner membranous layer in the mitochondria. What is the function of the mitochondria?
Powerhouse of the
Powerhouse. I like that. What do we mean by powerhouse? You're right in that it produces
the ATP.
Right, there you go. It produces the fuel for the cell to do work. And of course, that fuel, as mentioned, is ATP. Of course, you're not going to write ATP on the exam. You're, of course, going to write out adenosine triphosphate. We need to know how it does it. It actually does it in a process we call cellular respiration. And we'll see why it's called cellular respiration in just a minute. The job of the mitochondria is to completely break down sugars. But it can also do this to fats as well. So we have a sugar. However, if we want to break that sugar down completely, we need something special. What's that something special? We need to completely break down that sugar.
Like lysis?
Well, there are enzymes and things like that, but what's the other component that is needed? What's the other fuel source? What's the other thing that was needed? There you go, oxygen, excellent. Oxygen. We need oxygen to completely break down that sugar. When we do that, what happens is we get a massive amount of energy, and that energy, oops. Yeah, that was right. I know it wasn't. Energy. That energy is going to be used to make our ATP. From that, we also get waste products. One of the waste products is water. And what's the other waste product? CO2. There you go. Carbon dioxide. Again, this production of ATP, we use that energy from this process to make ATP. This cellular respiration is the whole reason you do this. The whole reason you is to get the oxygen in that our cells need and get rid of the CO2 that they're producing as waste. So the whole reason you do that is so that we can make ATP so that our cells can do their work. Mitochondria are really interesting organelles. One of the things that is unique about them is that they contain DNA. I guess that's not unique. Our nucleus does that as well. And notice both our nucleus and our mitochondria have double membranes. Both our nucleus and our mitochondria contain DNA. But they contain different DNA. They contain mitochondrial DNA. And mitochondrial DNA is basically the instructions for making mitochondria. Now, it has DNA in it. We can use it for identification. But mitochondria we only get from our mothers. We've already talked about the puzzle pieces, the egg and the sperm, right? And these are not quite to scale, right? We have the egg, we have that tiny little sperm, and that sperm is basically just a chromosome missile. That's a great question, Daniel. I'll answer that in just a second. That chromosome missile, all it contains is DNA. The chromosomes necessary to make that zygote. So basically all of the cell structures on the inside, the rough endoplasmic reticulum, the Golgi apparatus, the lysosomes, the mitochondria for that baby all come from mom. So one of the ways you can identify people is by using mitochondrial DNA. Mitochondrial DNA is actually easier to get and to synthesize. So often when people are finding cells and using those cells to identify people at crime scenes and things like that, they actually use the mitochondrial DNA for that. However, right, since it only comes from mom, is Maury using mitochondrial DNA to determine whether or not you're the father on his TV show at 2 o'clock in the afternoon? No, for that, obviously, he needs to use nuclear DNA because mitochondria only come from mom. For those of you not familiar, what Daniel is talking about, the symbiotic theory, is there's a theory that mitochondria are basically extracellular organisms that lived outside of the cell, that from a cell. So you had a mitochondria as a cell, another cell. They came together, kind of like peanut butter and chocolate. They came together and formed magic mitochondria. It was beneficial to them in that they were able to receive the nutrients and everything they need to survive, and they were able to provide more efficient energy for the cells to be able to grow and metabolize and function much more easily. I don't have a problem with that symbiotic theory. I think it seems reasonable. Obviously, I'm not well versed on the research to say one way or the other, but I think it is definitely a reasonable explanation of how that could come together. We've certainly seen lots of other microevolutionary examples of those types of things happening. So I don't know why that couldn't have happened way back in the day. All righty. So there you go. I think we have all of that. And again, I've got it with all the pretty words here as well. Double membrane, inner folded membranes of the cristae, cellular respiration, production ATP. Again, they have their own DNA, so they self-replicate, and that DNA just comes from mom.
Question.
Yes.
How did you define cristae again?
Cristae is the folds, the inner folds of that inner membrane. Let me say it that way. The folds of the inner membrane of our mitochondria.
Thank you.
Yep. All right. And with that, we are done with all of our organelles. Your book's got a nice table that talks about the organelles and does a good job of describing those. We still have to start talking about mitosis. So we have time for one more break. It is 11:23, so let's come back at 11:38, take one last 15-minute break. That's what I want. So we will return. Oops. At 11:38, is that what I said? Yeah. 11:38, and at that point, we will do mitosis, our first major physiological process. And I will restart recording at that time. All right. Any questions on that? All righty. Let's go ahead and get started. Any questions before we get rolling? All right, so like I said, we are now getting ready. We've talked about anatomy of the cells. It is time to start talking some physiology. And like I said, our goal is to talk about our first major physiological process, and that is going to be mitosis. However, before we can understand mitosis, there are a couple things that we need to talk about first. The first is our cell cycle. our cell cycle, and of course the key word in that, whoops, wrong button, I need that so I can bring up this. The key word in cell cycle is, of course, cycle. What do I mean when I use the term cycle? What does cycle refer to?
The path process?
True, absolutely. It is definitely a path and process, but what's the other key to it? There you go. It is a continuous, right? Something that is a cycle is something that is continuous and continues to go around and around and around. In a cell cycle, basically a cell has two major states in its life. Two major states in its cell cycle. And those two major states, or again, we can also use the term phases in its cell cycle, are interphase and the second one is the mitotic phase. So of course we need to define these phases, and let's start with interphase first. What is a cell doing during interphase? Anyone have any idea what a cell does during interphase?
Cell division.
Yeah, excellent. Well, not the division, although I like what Alina's put here. Really, interphase, two things are occurring. Those two things that occur, the first one is that the cell is doing all of its normal cell functions. So if it is an eyeball cell, it is doing eyeball things. If it is a toenail cell, it is doing toenail things. If it is a liver cell, it is doing liver things. If it's a gland, it is producing secretions, right? It grows and carries on all of its usual activities. However, at the same time it is doing all of its normal functions, it is also preparing to divide. So aside from doing all of its normal cell functions, it is also preparing to divide. And as you can see from this illustration, there are three stages or steps in this preparation. The first of these is what is known as the G1 phase. This one, as you can see from the illustration, is the most variable in time. Notice, as the illustration says, this process can occur in as little as 8 hours. But it also can take days for this process to occur as well. As you can see, during G1, one of the major things that the cell is doing is growing and making more cytoplasm. Remember, if one cell is going to become two cells, both of them need to have enough Golgi apparatus to survive. Both of them need enough rough endoplasmic reticulum to survive. So the cell is getting bigger and it's duplicating its organelles and it starts to make lots of proteins. Because division is a very, very special process, and that special process requires a lot of stuff. And like we said, it doesn't have attics where you can store those Christmas decorations in. We have to make them from scratch every single time. Another important thing that starts in G1. Notice it does not finish here, but it starts in G1, is the replication of our centrosome. As we know, our centrosome is the microtubule organizing center. As we mentioned, those centrioles of the centrosome is vitally important for cellular division. Remember, if we destroyed it with a laser, our cells don't divide. So having those two copies of the centrosome is vitally important. And here in G1, we start to replicate it, but we don't finish it yet. All right, questions on that? I'm going to cheat and move this stuff up so I have a little bit more playroom. Excellent. All righty. The next phase in interphase is S phase. S phase stands for synthesis. And any idea what we might be synthesizing? What are we making a copy of? DNA, absolutely. Of course, we know the DNA doesn't hang out by itself. It has those proteins in it as well. But here, we need to replicate the DNA, right? It's like having a deck of cards. I have a deck of cards, and it has 52 cards in it. If I and my sister both want to play cards, we could play with the same deck, but like I said, it's my younger sister. And older siblings don't want to play with their younger siblings, so it's much better to give her her own deck of cards so that she can play and I can play with my own. However, I can't just take my deck of 52 cards and give her 26 and me 26. None of us are going to be able to play a game that way. So I need to make copies of all the cards so that she gets a copy, I get a copy, and at the end, we both have a full deck of cards that we are able to use. And that's what we need to do here. If one cell is going to become two cells, then we need two copies of all the DNA so that both cells get all of the DNA so that they can be fully functional. All right. Once we've replicated the DNA, then the cell enters the G2 phase. In the G2 phase, this is where we make all the special proteins that are needed to divide. Lots and lots of massive protein synthesis is going on during this phase because we need all these special proteins and enzymes and other things to help in the defining process. And at the end of G2 is when we complete the replication of the centrosome. And in fact, completing that centrosome is then going to be the trigger that ends interphase. And when we end interphase, we then finally enter into the mitotic phase. So there you go. That is interphase. Those are the three phases of interphase, what happens in interphase, and what happens in each of those three stages of interphase. Questions on that? No questions? Excellent. All right. Then, if we are done with interphase, let's talk about mitotic phase. Step 2 is the mitotic phase. And interphase had 3 stages. The mitotic phase has 2 stages. Right, so there's 2 big differences. First, it only has 2 stages, and the other big difference is the 2 stages overlap. Notice with interphase, each stage G1, S, and G2 is independent, and as the arrows kind of indicate, there is a gap in between. So G1 occurs, then there's a period of time, and then S, and then there's a period of time, and then G2. Whereas here in the mitotic phase, we have 2 stages, and those 2 stages overlap. All right. Now, here is a classic example of, as I've said many times, I'm not sure if I've said it yet in this class, if I've had this rant yet in this class, but one of the important things that you will learn, if you learn nothing else in anatomy and physiology in 430 and 431, the most important thing you could learn is that anatomists hate you. Right? Absolutely. They're the worst people. And there are lots of ways that they give us examples of this. And this is a perfect example. This phase is called the mitotic phase. They could have called it cellular division. They could have called it all sorts of different things, but they called it the mitotic phase. And the reason they called it the mitotic phase is because one of the things that occurs in the mitotic phase is mitosis. But mitosis isn't the only thing that occurs in the mitotic phase. There is a second process that occurs in mitosis, and that is cytokinesis. So they have very, very poorly named this phase, right? Because mitosis is the rock star. It gets all the credit. It gets with the named after it. But, right, cytokinesis is just as important. It's kind of like Hootie and the Blowfish. Hootie gets his name, and then everybody else gets thrown into Blowfish, right? Mitotic phase is named after mitosis, but cytokinesis is just as important. And I know most of you didn't get the reference, and most of you think of it as a country star now. I don't care. All right, excellent. But what we do need to do is define them. What the heck is mitosis? Let's define these two stages. What is mitosis? Does anybody actually know what the actual definition of mitosis is? I like that, although I'd be careful about creation because remember, we're not creating anything, we're making things of it. So I like that. Let's combine what you guys said together, absolutely. The goal of mitosis is to take one nucleus and divide it into two nuclei, right? Remember, it's not cellular replication. We've already replicated the DNA. We've made all of the organelles. All of that stuff happened during interphase. So here, we're taking the two copies of the DNA and we're separating them into two different nuclei, right? We already have two copies of the DNA. So all we're doing is splitting those two copies into two nuclei. That's mitosis. Mitosis, in simplest form, is division of the nucleus. So then what the heck is cytokinesis? I like that. I like both of those phrases. That's excellent. It's division of the cell. It's the physical act. Cytokinesis is actually the physical act of dividing the cell, the one cell, into two cells. Or, again, the other way you guys can think of it is if mitosis is the division of the nucleus, then cytokinesis is division of the cytoplasm. Excellent. I like that. Perfect. All right. And again, well, we'll get to that in a second. One last bit. Mitosis, and notice I've left some space in here. Let's change colors of this just to emphasize it. Mitosis itself can be divided into 4.5 different stages. And those 4.5 stages have names to them. What are the names of the 4 stages of mitosis? Like you're not sure, you can cheat and look at the picture.
Prophase,
Absolutely.
metaphase, anaphase, and
there
telophase.
you go. Prophase, metaphase, anaphase, and telophase. And again, when it's right here on the screen in front of you, that makes it a little bit easier. But if you need a good mnemonic to help you to remember them and remember them in the correct order, just remember what you have in the bathroom. Because all of us in the bathroom have a PMAT, right? Prophase, metaphase, anaphase, and telophase. And notice as we were talking about They overlap. So notice before mitosis ends, about halfway through halfway through anaphase, cytokinesis begins. And more accurately, cytokinesis can actually extend beyond telophase. So it can actually go beyond. It doesn't end at the same time telophase does. It can actually go a little bit longer than telophase as well. So these are overlapping processes, and we'll describe all of these things that are happening in them in a lot more detail in just a minute, but this is the basics of them. All right. Questions on that? All right, we're going to talk about this mitotic phase in much more detail, talking about all 5 of these stages and what's happening in all 5 of these stages. But before we do that, there's one more thing here on the picture that we need to talk about, and that's this little fella down here. This little fella down here is a phase in a cell's life that is outside the cell cycle. There is actually a potential stage. Let's actually say that. I like that too. Let's write that. Potential phase in a cell's life that is outside the cell cycle. And this phase outside the cell cycle is what is known as G0. Note, apoptosis is when a cell dies. In this case, the cell is not dead. The cell is not in hibernation or sleep or anything else like that. During G0, a cell is doing all of its normal functions. So eyeball cells are being eyeball cells. Toenail cells are being toenail cells. Liver cells are super active, making bile and filtering blood and doing all sorts of other things. They can be super busy and super active and absolutely positively not dead. So what the heck is the difference between G0 and interphase? Because didn't the cell do all of that things in interphase? It did. Cell does all of its normal functions in interphase. So what the heck's the difference between G0 and interphase? There you go. Excellent. It's not so much that it kickstarts interphase, but you kind of get the right idea. In the G0 phase, the cell is doing all of its normal cell functions, but it is not preparing to divide. And absolutely, all of these stages, every single one of these stages, how long they are, is determined by what type of cell they are. Absolutely. And again, that's absolutely true for G0, but it's absolutely true for all of these phases. Right? And that's the key. Most cells spend at least some time in the G0 state. Now, of course, 2 key words in that sentence. Most. What does most mean?
Not all.
Not all. Excellent. And at least some means that how much time could vary. So let's think about a simple state. What kind of cell do you think would get done dividing? And as soon as it got done dividing, it would either pay very little time in G0, or maybe it would go right back into getting ready to divide again. What type of cell do you think would spend little or no time in a G0 state?
Zygote?
Zygote would definitely be one, but a zygote is really just a special example of what?
Stem cell.
Stem cells. Excellent. Right? Remember, he said stem cells, their job is to divide rapidly. So they aren't going to spend very much time in the G0 state, like, for instance, the skin cells on your skin, Or like the cells on the roof of your mouth. You had that nice big scalding hot cup of coffee this morning and it burnt the roof of your mouth because, again, I was yammering on for hours as usual and you were exhausted and you were trying to do something to wake you up. And now you've burned the roof of your mouth and it's a little painful and it's a little sore and it's a little red. But by tomorrow morning, it's pretty much going to be better again, right? Those cells divide super rapidly and they repair themselves super, super rapidly. But what if instead of hot coffee in the morning, this class causes you to drink vodka for breakfast, lunch, and dinner every day for 3 straight weeks, right? At that point, you're going to start damaging your liver. And 3 weeks become 3 months, and 3 months become 3 years. And after 3 years of having vodka for breakfast, lunch, and dinner, you've done some serious damage to your liver. Now, if you stop drinking, can your liver repair itself?
Yes.
Maybe. Yeah, maybe. It depends on the extent of the damage. Because here's the deal. Yeah, there's the key. Slowly, kind of. Your liver cells, once they get done dividing, they enter this G0 state and they stay in the G0 state for about a year. So they go into this G0 state where they're doing all of the active stuff that your liver needs to have done. And then after about a year, they start to get ready to divide again. Right? Absolutely. As Daniel pointed out, typically now, when you get a liver transplant, you no longer get a whole liver. You get a portion of a liver. So they take half of the liver out of somebody else and they give half of it to you. And so for a while, you guys both have half a liver, which is okay. It can still do everything that the liver needs to do. But 10, 15, 20 years from now, you're both going to have full-size livers inside of your bellies. Because eventually it's going to grow back. But not over the night, not over the weekend, not over a year. It's going to take decades for it to fully form back out because it is a slow growth process. So whereas your skin grows back very rapidly, your liver not so much so because they spend different amounts of time in this G0 state. And off the top of my head, I can think of 2 examples of cells that once they mature, enter the G0 state and never leave it. What happens if you enter the G0 state and never leave it? If a cell enters the G0 state and never leaves the G0 state, what can that cell never do? There you go. No more mitosis. It can no longer divide. And if we repair tissues by replacing them with damaged cells and our cells don't divide to make new cells, can you repair that tissue?
It's scars.
Yeah, you get scar tissue instead. The tissue doesn't repair itself. So what are the 2 types of tissues that I thought of right off the top of my head that you know when you injure them, they can't repair themselves? I know you guys know the answer to this. You're overthinking it. Nervous tissue, excellent. Nervous tissue is one of them. You get a traumatic brain injury. Like I said, you play Superman in a movie and hurt your spinal cord. You spend the rest of your life in a wheelchair. And there you go. Cardiac muscle is the other one. Cardiac muscle doesn't regenerate. Nervous tissue doesn't regenerate. Because those cells, once they're mature, they enter the G0 state and they never leave it. They never divide again. All right? Questions on that? All right, excellent. Okay, so we need to talk more about mitosis and cytokinesis. However, before we do that, we need to talk a little bit more about interphase. And in particular, I want to talk about this process here, the S phase, the replication of our DNA. I promised you when we talked about DNA that we would get more in depth of it. And this is our chance to do that here. We did all that. We did all of that. Excellent. If you remember, and here's a good reminder of some of the information we started with this box right here. We know that nucleic acids are made up of strings of nucleotides. Let's clear that. Let's write that. DNA is made of strands of nucleotides. Oops. Nucleotides have 3 components to them. A sugar, and remember when we're talking about DNA, which sugar is it again?
Deoxyribose.
There you go. Excellent, that deoxyribose. And if it was RNA, it would be ribose. A phosphate and a nitrogen base. And remember, we said that there were 5 possible nitrogen bases: adenine, guanine, cytosine, thymine, and uracil. And we hinted at it, but we didn't talk about it too much. These come in 2 classes of nucleotides. We have purines. These are the larger of the nitrogen bases. Notice they're made up of 2 nitrogen rings. And there are 2 purines, adenine and guanine. Pyrimidenes are the smaller, larger name, but smaller shaped, and they are only made up of 1 nitrogen ring. And there are 3 of these: cytosine, thymine, and uracil. However, as our illustration here shows, both DNA and RNA each only use 4 bases. Both DNA and RNA use adenine, guanine, and cytosine. However, DNA only uses thymine and RNA only uses uracil. So if we're talking about DNA and DNA replication, the only bases we are going to have are A's and G's, C's, and T's. All right. Now, we talked something else, and let's go back to our pretty pictures to do this here. Clear that. As we talked about, oh, let's cheat. Our sugars, and I'm just going to put boxes for sugars. And our phosphates. Are connected together to each other to form a phosphate sugar backbone. And off of this backbone is where we find our nitrogen bases. And as I mentioned, and again, I'm just going to continue to use the letters because it's easier. They can be an A, they can be a T, and actually, oh, not too late now. T, they can be a C, and they can be a G. However, DNA comes in a double helix. Basically, it is 2 strands of DNA that wrap around each other. Now, I can't make it wrap around each other very nicely on this, so I've got mine straightened out. But the key is, over here on this side is going to be a 2nd strand with its deoxyribose sugars and its phosphates. We're not going to worry about 3' and 5' and all of that. I know you'll get that in micro, so I don't care about that here. It's the beauty of having this and micro being most of your responsibilities. All the stuff in micro that is either made up or I don't care about, I don't have to talk about because I know you guys will get it there. It's so awesome. Excellent. And we're going to have another set of bases here that are going to be connected to and influenced by the other. However, as many of you know, when we're forming these complementary pairs, not any old, actually we should change this around. Hold on. Let's put the two purines together and the two pyrimidines together. Excellent. I like that better. All right. Not just any old nucleotide can pair with A, for instance. So remember, these two here are our purines, the large ones. And so large always pairs with small. So can either C or T be over here as the complementary pair to A?
T.
T, absolutely. Haley already cheated, and while I was rambling on, wrote it all out there efficiently for me, so thank you for that. Absolutely. A only pairs with T, and C only pairs with G. So, of course, what would go here?
G.
G. And what would go here?
A.
A. There you go. So A always pairs with T, C always pairs with G, and vice versa. Those are our complementary base pair rules. Now, the reason for this, we'll see, is because of the structure of them. Oops. Oh, there it is. Notice we have our two large ones, the purines, and our two small ones, the pyrimidines. And notice adenine and thymine form two hydrogen bonds with each other. So two hydrogen bonds form to hold them together, whereas both guanine and cytosine can form three hydrogen bonds. So three hydrogen bonds hold the C's and the G's together. Two hydrogen bonds form the A's and hold the A's and the T's together. And that's why A can't go with C and T can't go with G. Yep, so as we put this together, and notice, again, I did an amazing job of drawing it, but let's look at the pretty picture from your textbook because it does a nicer job. Notice here, when we look at the nicer job, we can see this strand over here represents, oops, I want that, represents the sugar phosphate backbones, and here we see them wrapping around each other. And then here's the second strand. And notice, using our complementary base pair rules, A forms two hydrogen bonds with T, G forms three with C, and then T with A, and C with G, and C with G, and A with T, and G with C, and T with A, and A and T, and C with G, and so on and so forth. Notice we can simplify this a little bit by making just a simple strand. If I had a simple strand of DNA, C, A, T, oops, T, G, G, C, T, A, and I had that simple strand of DNA, could you tell me what the complementary bases for this would be? Yeah, absolutely. So what would go across, what would pair with the C?
G.
G, excellent. And the next one?
T.
Don't type it. I can't type. I can't look, type, and see what I'm doing at the same time. So you guys got to say it out loud. T, what's next?
A.
A.
C.
C. Don't make me say what's next each time. C.
G.
G.
A. T.
A. T.
T. Bingo. It's just that simple. And in fact, when it comes time for our cell to make new DNA, that is exactly how it does it. Here is a strand of our DNA inside of our cell. And remember, what's holding these two strands together? What's holding the two strands together?
Hydrogen bond?
Hydrogen bonds. And are hydrogen bonds really strong or really weak?
Weak.
Weak. Absolutely. So what our cell does, using enzymes, it pulls the two strands apart. And once it pulls the two strands apart, each one becomes a template. What goes here? What goes across from the C?
G,
G. And then?
T,
And then?
A,
Don't make me keep saying and then.
B,
B, C, G,
A, T.
Excellent. And we do the same thing over here. What goes here?
C,
Oops.
A, T, G, G,
G, C,
E, T, A.
Excellent. And notice now what we have, where we had one strand of DNA before, we now have two identical strands of DNA, one part of the original and one a new one. That was pretty easy to do, right? Well, that's exactly what the cell does. When it comes time for the cell to divide, what you just did right here is exactly what the cell does. It peels apart the two strands of DNA and uses each one of them as a template to make a brand new strand. So now I have two identical strands of DNA. Each one has an old strand, each one has a new strand, and they're perfectly identical. So one cell can get one and the other cell can get another. Now, is it truly just this simple in real life? Yes, but it requires a tremendous amount of work. There's dozens of proteins and dozens of enzymes and all sorts of other things and double checkers and all this other kind of stuff that goes on. Like I said, I know you'll talk about this in more depth in micro, but what I want to talk about is that when it divides, the primary protein, the primary enzyme involved in this process is an enzyme by the name of DNA polymerase. DNA polymerase, wowzers, is the primary enzyme that is involved in this process. It plays a role in separating the DNA and using that complementary base pair rules just like you did. And when it sees a T, it adds an A. When it sees a G, it adds a C, and so on and so forth. Now, again, it's not the only one. Notice here we have helicase that is helping to unwind it. There are others that stabilize it. Like I said, there are others that double check it. There are dozens of these again. And remember, A, T, C, and G are four nitrogen bases. And notice, conveniently enough, they have the copies of them down here: adenine, thymine, cytosine, and guanine. All right. And these are what put these things together. So the DNA polymerase is the primary enzyme that does exactly what you did. Using complementary base pair rules, makes a new strand of DNA. All right, go back to here. This is what you wanted to see? Allison, is this what you wanted? All right, excellent. All right, and again, since Laura asked the question, we'll go back here. Remember, Laura, these are our nitrogen bases. So adenine and guanine are the nitrogen bases of our nucleotides. They're the purines, they're the large ones, the two nitrogen rings. Cytosine, thymine, and in RNA uracil are the pyrimidines, and they are a single nitrogen base. Those are the nitrogen bases. All right, excellent. We now know the basics of the process of DNA replication. So what we need to do now is we need to talk about the process of mitosis and moving those chromosomes around. Now, as I look at the clock, I see that we have run late. This is a long process that we can't get into with 15 minutes left in the class. So what I'm going to do is go ahead and stop the lecture here. Even though we're a bit behind, I want to make sure that we understand this concept. So what we're going to have to do is save this for Wednesday. On Wednesday, we're supposed to start the plasma membrane, but we will start here first. So we will start here first with finishing off mitosis. And then from there, we will go into the plasma membrane. All righty? Any questions on any of that?
Sir, what did you say? On Wednesday, you said when after finishing this lecture, what are we gonna do?
Well, we're going to go straight into the plasma membrane, talking about the structure and function of the plasma membrane. All righty. Now, your mitosis, even though we didn't get a chance to talk about this in class, your mitosis labster is still due Wednesday. So make sure you get that done. And the good news is that means that Wednesday's lecture is going to go a lot smoother because you'll have already looked at and understand these concepts. All right? Questions on that? All right, that is all we are going to do for today then. I thank you guys for a fun lecture. I always like the cells. This is fun stuff. And then we will move on from here. Yes, if you did not get 80% of the labster, you can do 80% correct. You can redo the labsters. That is correct. But as long as you get 80% correct, you will get full credit. And like I said, I have updated those on Canvas already, those grades. All right. Any other questions? All right, excellent. You guys have an excellent day, and I will see. Yes.
For the review, for the first review, it's also like 80%. If you're 80% correct, so you will get the full credit for that?
No, remember the unit reviews, I'm just looking for completeness. I am looking for time and effort. Technically, if you put time and effort into every single question, you have good answers written out, good explanations for your answers, and every single answer is wrong, you can still get full credit. Now, if you do the unit review and get every single question wrong, I'm worried about you when it comes test time, but at least you've been trying to study, and so technically you would still get full credit for that.
Yeah, thank you so much. The only thing, the reason why I asked this question, that the last question we had some cross sections.
Mm-hmm.
Cross sections, like the first cross section, the third one, the A, I could not get the meaning of that. Like I didn't understand like which section is this. So I did the two, like the posterior and anterior, like the mediastinum and also the spinal cord and the right and left pleural cavity.
Mm-hmm.
So I identified all those, and the first one, like I was not sure that what should I write for this one.
Well, so that would be the perfect thing to come to my office hours and ask me about if you were confused about an assignment. Sure. Okay?
Thank you, sir.
Yep. All right. Any other questions? All right. Then, like I said, you guys have an excellent day, and I will see you bright and early Wednesday morning.
Okay, thank you.